Days International Conference on Emerging Trends in Medicinal and Green Chemistry, sponsored by Union Bank, SPA Cure Labs, Touching Lives Globally, and Jolly Scientific Traders. Science knows no country because knowledge belongs to humanity and is the torch that illuminates the world. It's a quote by Louis Pasteur. So here I am, Nazia Fatima, Assistant Professor, Department of Chemistry. Extend a warm welcome to all the dignitaries, esteemed speakers, the chairpersons, participants around the globe, and my dear colleagues to the inaugural ceremony of the 2-day international conference. Let's begin the day's proceeding, invoking the blessings of God. So, may I request Afia Mary Ma'am, Head, Department of Chemistry, UG, to lead.
The scripture portion for the first day of conference is taken from Psalm 118. Give thanks to the Lord, for he is good. His love endures forever. It's better to take refuge in the Lord than to trust in humans. It is better to take refuge in the Lord than to trust in princes. I will give you thanks, for you answered me. You have become my salvation. The stone the builders rejected has become the cornerstone. The Lord has done this, and it is marvelous in our eyes. The Lord has done it this very day. Let us rejoice today and be glad. Lord, save us. Lord, grant us success. May Lord bless this portion for us.
Purify my heart, let me be as gold and precious silver. Purify my heart, let me be as gold, pure gold. Refine us fire, my heart's one desire is to be holy, set apart for you, Lord. I choose to be holy, set apart for you, my master, ready to do your will. Purify my heart, cleanse me from my sin, and make me holy. Purify my heart, cleanse me from my sin deeply. Refine us fire, my heart's one desire is to be holy, set apart for you, Lord. I choose to be holy, set apart for you, my master, ready to do your will. I choose to be holy, set apart for you, my master, ready to do your will, ready to do your will.
Thank you, Afia Ma'am, and dear students.
Thank you,
Now,
Nancy.
may I request Dr. Sister P. Amruta, Principal, St. Anne's College for Women, Dr. Smita Asthana Ma'am, Dean, Academics and IQAC Coordinator, Dr. Amani Das Ma'am, Convener of the conference, Dr. K. R. Radhika Ma'am, Honorary Secretary and former Honorary Secretary, RSC, Local Section Deccan, and former Head Department of Chemistry, Vijayvalli Ma'am, Additional Controller of Examinations, to inaugurate the conference by lighting the lamp. Requesting Amreen for the digital display.
शुभं कुरुत्वं कल्याणम् आरोग्यं धनसम्पदः शत्रुबुद्धिविनाशाय दीपज्योतिर्नमोऽस्तु ते दीपज्योतिर्नमोऽस्तु ते दीपज्योतिर्परब्रह्म दीपज्योतिर्जनार्दनः दीपज्योतिर्परब्रह्म दीपज्योतिर्जनार्दनः दीपो हरतु मे पापम्
Thank you, Dr. Amreen. It's a great honor for me to welcome our Honorable Principal, Dr. Sister P. Amruta, St. Anne's College for Women, Chief Patron of e-Conference, a dynamic leader and an inspiration to many, with the virtual green greetings as a token of gratitude. May I request Dr. Sister Principal to address the gathering and declare the conference open. Over to you, Sister.
Good morning, everyone. It is with great pleasure that I welcome you on behalf of St. Anne's College for Women, Hyderabad, to the International E-Conference on Emerging Trends in Medical and Green Chemistry, organized by the Department of Chemistry. I extend a warm welcome to Dr. Ravi Shukla, Associate Professor, School of Science, Cluster, Biosciences and Food Technology Department, RMIT, Melbourne, Australia. Welcome, sir. Dr. Kalpana Madugula, Research and Development Chemist, SAS, Nanotechnology, USA. Welcome, ma'am. Dr. Ravi Challa, Senior Professor, Sumaninen, Finland. Welcome, sir. And Dr. Shanti Priya Samudrala, Assistant Lecturer, Department of Chemical Engineering, Faculty of Engineering, Monash University. Welcome, ma'am. It is an exciting day and an opportunity to learn from resource persons with such diverse experience and expertise. Born in 1983 in a two-room college with 106 students, St. Anne's has emerged as a leading institution with 35 program options and 3,500 plus students. Beside pursuits of academic excellence, the college imparts education with the mission of transforming its girls into empowered women who are socially conscious, intellectually competent, morally upright, and physically healthy. The accreditation by NAC with A plus grade and the status of college with potential for excellence by UGC twice in 2010 and 2014 is a manifestation of our commitment towards quality education. Our institution is also recognized as a mentor institution under Parmasha scheme of UGC during the current year. Our strategy to accomplish the mission of the college is rooted in creating a favorable academic environment to promote teaching, learning, and research, monitoring the students to make quality an integral part of the institution, and motivating commitment and dedication. Presently, all communications, interactions, and deliberations have moved to virtual platform, but what remains rooted is our commitment to provide quality education with guidance and mentorship from the academic community. To safeguard the environment, green route of synthesis has gained significance and medical chemistry is need of the hour. The management and faculty of St. Anne's has always been ready to explore changes and challenges, and this event is a small step in that direction. At this unprecedented pandemic times, emerging of new trends, new solutions are very much needed. I am happy to note enthusiastic participation from researchers across country and abroad. I congratulate Department of Chemistry for this initiative and wish all the participants an enriching learning experience. I acknowledge the support provided by Union Bank of India, SP Acure Labs, and Jolly Scientific Trades. All the best. Have a nice time. Thank you. God bless you all.
Thank you, sister, for enlightening us. I take this opportunity to welcome our next dignitary, Dr. Smita Asthana Ma'am, Dean Academics, Reader in Chemistry, and IQAC Coordinator, who's a role model, St. Anne's College, with the greetings.
Thank you, Nasia. It's so beautiful.
Thank you, Ma'am. May I request Dr. Smita Asthana Ma'am to address the gathering? Over to you, Ma'am.
Thank you, Nasia. It's a pleasure to speak and represent my department. So, a very good morning. A bright day with sunshine and health for all is what we seek today. A very hearty welcome to fellow chemists. Everybody is a chemist. We are at chemistry department. We are a very young department, just 38 years of age. We started with one UG program of BZC with 60 students and one lab. And today we have grown to 9 program options. We have 10 labs. And at any given time, we have 1,000 plus students at undergraduate and postgraduate programs. We are a good mix of youth and Isaac Newton kind of hair. Department has a number of research projects supported by UGC, TS Cost, ICSSR, and industry and other bodies. Students are also encouraged to take a project work along with faculty. With number of books authored by department members, publications in peer-reviewed journals, and few patents have also been filed. Last year, March, exactly today, did we even think for a second that what was in store for the whole world? How time has totally changed, the word order has changed. In fact, extreme cooperation, sharing of data, research findings, empathy, and economic tumble, all this happened together. We are still accepting new normal. We have witnessed the importance of collaborative research, and a conference idea was born to introduce young mind to research more than ever. Of late, green route to synthesis and eco-friendly mechanisms have gained significant interest among researchers in industry and academia. Developing and discovering new drug molecules, therapeutic agents for curing disease is a continuous sustained process. So this conference, Emerging Trends in Medicinal and Green Chemistry, aim to merge this both significant genre and provide a common platform for researchers globally to showcase their significant findings and development. Also, it connects people across globe to share their knowledge and benefit from each other with solving problems related to medicine development and alternate synthesis. I'm happy to share with you that A good number of abstracts were received, and after careful scrutiny by review committee, a few were selected for presentation. And the best presenters will be awarded a token prize, and selected papers will be published as book chapter with reputed publication and our online journal with impact factor. So very much looking forward to new learning. I once again welcome all resource persons, research scholars, faculty and students. Let's get set and go. All the best. Thank you.
Thank you, ma'am, for the detailed information about our department, Born with the Best. I am pleased to welcome and introduce our convener, Dr. Amani Das, ma'am, Head, Department of PG Chemistry, with the greetings. May I request Amani Das, ma'am, to give the opening remarks. Over to you, ma'am.
Thank you, Nazia. Good morning, everyone. On behalf of the organizers of ETMGC, I take pleasure to invite all the delegates, guests, and participants present here today. This two-day conference is having 4 sessions led by eminent people from different national and international places to talk about the upcoming trends in medicinal and green chemistry. Hope this conference promotes top-level research and other global aspects through participation and paper presentation. I am thankful to everybody for the, for the overwhelming response for the paper presentations. We hope that this conference emerges into a very helpful technical session for everyone present today. We are looking forward for a great session during these 2 days. Thank you.
Nazia, you have to unmute yourselves.
Oh, regret for the inconvenience. Now it's time for e-release of souvenir by the dignitaries. Dr. Sister P. Amrita, Principal, Dr. Smita Asthana, ma'am, Dean Academics, Mrs. C. Vijayvalli, ma'am, Additional Controller of Examinations, Dr. Amanidas, ma'am, Head Department of PG Chemistry, Miss Afia Mary, ma'am, Head Department of UG Chemistry, and Dr. Amreen as the convener. Over to you, Amreen, for the virtual release. Thank you, Dr. Amreen. I now invite Ms. Ramya, Assistant Professor, Department of Chemistry, to introduce and welcome Dr. Ravi Shukla Sir, Associate Professor, School of Science Cluster, Biosciences and Food Technology Department, RMIT, Melbourne, Australia. Over to you, Ms. Ramya.
Good morning, everyone. I hope I am audible.
You're audible, yes.
Yeah, I feel extremely delighted and honored to present a brief profile of speaker of first session of ETMGC 2021. So when I read your profile, I was just wondering how 2 minutes would be suffice to include numerous scientific research contributions you have made. Well, here is the inspiring profile of Professor Ravi Shukla. Ravi Shukla received PhD in nanobiotechnology jointly from National Center for Science, DPT, and National Chemical Laboratory, CSIR, India in 2007. After PhD, he chose to work at National Cancer Institute's Cancer Nanotechnology platform at University of Missouri, USA, and worked towards the development of promising nanotech therapies and diagnostics. In 2011, Dr. Shukla moved to Australia as Vice Chancellor's Research Fellow to join Advanced Materials and Nano Devices Research Program at the School of Science and RMIT University, Melbourne, Australia. He is currently an associate professor there and leads targeted theranostics and biosensing research at RMIT. Professor Shukla has made significant original contributions in the field of nanomedicine. He is a celebrated scientist in the field of nanobiotechnology, cell biology, targeted theranostics, biosensors, and tissue engineering. Professor Shukla has authored over 140 research publications and over 20 patents have been filed and for his invention disclosures. His research has been cited more than 5,800 times and his H index is 34. He is a pioneer in the field of gold nanoparticles and its biomedical applications, which span imaging, therapy, diagnostics, and biosensing. His seminal work, published in Langmuir, has attracted more than 1,670 citations. To mention, this finding for first time provided scientific evidence that nanoparticles of gold are biocompatible, which is now universally accepted and regularly employed in a range of biomedical investigations. Sir has devised specific protocols for nanoparticles, bioconjugation with receptor-specific ligands, and has established greener protocols for synthesis of nanoparticles of different shapes, sizes, and chemical nature, which could be further exploited as imaging biosensor probes for myriad of applications. Two of nanomaterials which his team has developed are currently in different phases of clinical trials. Due to his multidisciplinary nanotechnology research expertise and international reputation, Dr. Shukla serves on several international academic committees which spans the globe and in editorial boards of reputed journals. He is a regular reviewer for over 15 international journals and serves as external reviewer for Australian Research Council ARC and National Health and Medical Research Council NHMRC research programs. He is an adjunct professor at Institute of Green Nanotechnology at University of Missouri, USA, and a visiting professor at IICT, India, and also at University of Padova, Italy. Now, I request Dr. Amreen to present virtual green greetings to Sir. Amreen? Sir, we hope and aspire for stronger and greener collaborations with you. We all are enthusiastically waiting for your talk. Over to you, sir.
Thank you so much, Dr. Das, for such a generous introduction for me. And a very warm greeting to all of you from Melbourne. And thanks to our organizing committee to inviting me for this talk. And I feel inspired, actually. The way you are running this show is great, commendable. So although I work in the area of nanobiotechnology, green word is implied. I have not emphasized more onto the green word in my presentation, but it's implied and you will get a sense of when I'm talking later on. So you will get a sense of like where green chemistry, and you can get these things in somewhere in the presentation. So here I am. So I wanted to give you an overview of today's presentation. Just wanted to introduce you with my university where I'm working currently. Then a bit, because I see lots of students in the audience. So as a brief introduction, very brief introduction to the nanobiotechnology. Then my current nanobiotechnology interest and research activities with two of the examples. So I'm going to put my clock on so that I am in time. How much time I have? around like okay, so I'll go with like I will try. Okay, so what I will do, I will try to be pictorial more, telling you a story, and then when you have a question, we can have some question and answer hour, right? So let's go. So this is my university. Is the in the heart of Melbourne city. If you come to a city, it's called Central Business District, and this is the main building into the heart of the city. So if you can see, this is the aerial view of my university, which is over somewhere here in the Central Business District. And you can note that here is a Melbourne cricket ground. So it's not very far. We can walk. It's not very far from my university. And somewhere here is the place for Australian Open tennis. So RMIT University is stands for like Royal Melbourne Institute of Technology. So it's more on technology and design university. We are the largest technical tertiary education in Australia, with 84,000 students enrolled in, and it's supported by 1.1 billion annual budget. Yeah, needless to say, because our most of the students are like around 35%, one third students are international. So we got a hit in this pandemic. What Sister Amrita was telling is, yeah, we could feel that what in the business. So by numbers, this data is a little bit old, but we are 81% higher education, and out of these, around 36% are international students. 16% of these are studying in Australia, and 20%, roughly 20%, are still outside the Australia. We have 5 campuses, 3 campuses in the Melbourne, and 2 campuses in the Vietnam. Apart from that, we have presence in Europe in Barcelona. We have RMIT Center, and then we have joint PhD program with the 14 organizations worldwide, including so the program we started with the Indian Institute of Chemical Technology, Hyderabad, which is now extended to Academy of CSIR. So we are now connected with all 40 labs from the CSIR India. This is because I work in the area of material science. Just wanted to give you a flavor of that. We have state-of-art laboratories with class 100 rooms, and where we can do multiple material science experiment. And this is in the heart of a School of Science, which is in the city. And this is a suite where we can do advanced microscopy and microanalysis, just because it is, these are the facilities which I use on a routine basis. And then the biggest emphasis is in my university is on the skill development. So my undergraduate students, they handle these advanced experiments, which are electron microscopy and XPS, such advanced they do for their project, and the PhD students, they do use these facilities by their own hand. So this is kind of like skill development is a big emphasis in my university. Just so if you happen to be in Melbourne, anyone, I want to invite you. Whenever you're coming to Melbourne, I will be happy to host you in Melbourne and then show you my university. If you want to, if you have interest in interacting, yep, you're most welcome. Please feel to write and get in touch with the organizing committee. They should link you with me. So when I'm talking about the nanoscale and how big is nanoscale, so I work in areas, this is a very large volume, and then you say if you take this size, so I work somewhere in between 100 to 1 to 100 nanometer sizes. And why this is because in this size range, you say most of the viruses and the proteins in our body. And so to give you a perspective, if this is a 100 nanometer size, the bacteria is around 10 times larger than this, and our red blood cells are over 70 times larger. So what I'm talking about somewhere here in sub 100 nanometer particles, which are, yep, so we have studied, we know very well atomic structures, but when it comes in nano, we see very exotic properties into the materials. So the area in which I work is called utilization of nanoscience in the biotechnology. So I use nanomaterials and nanoscience. I get inspired and I apply them into my biology or biotechnology because my basic background in biotechnology. So this is called nano biotechnology. Similar, there's a parallel branch which is bio nanotechnology, where we utilize the biological principle, biological science principle, get inspiration for nanotechnology. For example, DNA nanotechnology is a example of a bio nanotechnology because we use the DNA or these biomolecules as a template to synthesize materials, so that we can synthesize materials in the wire form and different morphologies. So just this lecture is a tribute to Professor Richard Feynman, who is considered as the father of nanotechnology. And then I want to take you to his lecture, actually, which he gave in the end of the 16th century, 16th decade, 1960. So what he is saying, and he's talking that time, actually, long back, he's trying to say that that as soon as I mention this, people tell me about miniaturization, making things smaller, and how far it has progressed. So they say they can write large square onto the pinhead, and then in 2000, he is predicting that in the year 2000, when people will look back to this age, they will wonder why people were not thinking about the nanotechnology that time. What I want you to concentrate on to the, when it was written, this is expert, you can see the width of the letter L over here, which is around 0.6 millimeter, which is 600,000 nanometers, right? Exactly what he predicted in 2002. Chad Markin's group wrote the same expert using a technique called lithography. And if you can see, concentrate onto the people L, how much miniaturization we could get in our ability. So you can see it's a 60 nanometer width. And same way, if you see a D direction in the D, it's a 400 nanometer. So it's written by lithographic techniques, and then you can see that we have achieved whatever he predicted in 1960s. That was exactly possible in 2000. Why size is important? It is because I will give you a very classical example, which is, suppose you have a big block of material or metal or something, where you have total number of atoms are around these, and then these are the numbers which are on the surface. When you divide it in 8, what you see, the number of total atoms are same, but the number of surface atoms are increasing. And then you further divide, so you can see the exponential increase into the number of surface atom. What is this mean? That the surface area. The surface area to volume ratio increases tremendously, and that's why these materials start showing exotic properties like. So at a nano domain, what happens? The fundamental properties of matter change at the nanoscale. The properties of atom and molecules are not governed by the same physical laws as larger objects or even larger particles. So these properties which are altered, they are I have listed some of these, but it's not limited to these. So you can see drastic change in the color of the material in a nanoscale, solubility, material strength, electrical conductivity. So what we started doing, we started to learn around 15 years back when I started my career. We started to think about how we can use these exotic properties at that time, because I was early adopter in India when I was doing my PhD. So the goal was like how we can use these material into the potential application which can be diagnostic imaging and drug delivery and therapies. So this is the example which I wanted to give you, and is seeing is believing. So what you see is a change in the size in the nano domain. So if you take the gold looks like a golden, but when it comes into nano domain, so this is around 25 to 40 nanometer particle size, which is ruby red in color. As the size increases, you see there is a change in optical properties of the gold nanoparticles, and then you can see this in the gold. You can see different type of colors coming, and these have lots of implication in the application side of. Similarly, this example is from the quantum dots, which are cadmium selenide quantum dots from the Cavendish group. So when the size changes in nano, small size changes also lead to much more differences into the properties of the material. So how they can be utilized? This is one of the example which I'm giving, where you can take different kind of ratios, and if you enclose them, you can see you get a profile of the materials which could be utilized for the barcoding on these kind of explanations. Just changing the ratio, you can get multiple profiles of materials which could be used for multiple different kind of applications. So another example is, which I wanted to give, is from our own, and I will emphasize because I will tell you the story of this one. That's why. So this is an example where we have synthesized material gold nanoparticles using green tea. So green tea has lots of antioxidant, and if you have a basic chemistry, you know that if you take a gold salt, because gold is an electrode in this position in the electrochemical series, it readily reduces. So the phytochemical cocktail within the tea or any other, we have tested many of these plant products and extracts. They have phytochemical cocktails which are able to reduce very easily gold, and then you can get particles which look different in color. And then this is electron micrograph. You can see these particles are very tiny particles. These have a very nice normal distribution in their particle size, around like 30 to 60 nanometer size. But if you see here, a subtle change in the profile of nanoparticles, you can see there are two optical signatures which are. And because of the gold, silver, and noble metal like platinum, they have a special property on the surface, plasmon resonance. Because of the SPR, you can see a peak of under UV-vis NIR spectroscopy. You can see a peak somewhere around 530 to 550 nanometer. These particles are quite stable because so normally if you take colloids and then you add a little bit of salt, they tend to aggregate. But in this case, these are quite stable particles. So we'll go back to these, but because of this change in batch to batch, you can develop. So undergraduate in the class, my class, they started using this profile as we use as a biosensor for the tea quality, because this is the antioxidant potential, which is so if you take a green tea and from different sources, what she did, easily got the green tea from the different sources and check the profile, and then the color of that indicated which is the good tea, because of the with high amount of antioxidants within the green tea. So that can be a biosensor for using green tea based quality of green tea. This is an example which is coming from our group. So there are many fascinating nanomaterials have been synthesized, and they are in literature, and different applications have been reported. Some of them are in clinical trial also. So these are most common of like type of nanomaterials which have been used and tried into biomedical applications. So how they can be synthesized? In general, there are two ways to synthesize. One is a top-down approach, where you chisel out a bigger material, you keep on beating them, and then ball milling, and then you can bring the size into very small. So this one example comes from the Swarnabhasma, actually, from the Ayurveda. If you see, the same, Many of you know that because the why Swarna Bhasma looks orange or reddish because of the particle size, which are very small in here. So that is a kind of a top-down approach, while another approach is which is a chemical approach use is a self-assembly approach where you can do bottom-up approach. So mostly what we are doing in the lab, we are using the bottom-up approach for synthesizing in our research. So there are many synthetic methods comprising physical, chemical, and biological ways of synthesizing these nanomaterials. And what we have tried to exploit the power within the plants, extracts, waste, and phytochemicals to come up with some usable form of materials which could be utilized for the biomedical applications. So these are the for the young people into crowd who have limited experience. So there are the tools and techniques. So this I would say this list is not exhaustive, but there are many tools and techniques applied into the characterizations of nanomaterials. So these are the tools which I routinely use in my lab to study materials. For example, visual appearance for the gold and silver. Silver is yellow color. Then you do spectroscopy, electron microscopy, dynamic light stacking, and these gives different information about the material as well as the composition. So it's just a very small kind of a brief introduction I wanted to give you. Now I will take you slowly what we do in our lab. So the research which I do is a kind of applied research where we have usually materials. We have studied metal oxide, metal organic frameworks. There are macromolecules, drugs, and the hybrids of these to synthesize nanomaterials. So this is the one example which is currently in the clinical trial for the imaging of the cells in vivo. So this is in first in human trial. I will tell you a little bit more about later on. So our ability is that we can synthesize the core and then bioconjugate these materials with different kind of ligands, or we can create porous materials where we can put drugs and biomolecules within the material framework, or we can do both way where these are inside and out with different kind of possibilities. So we have generated many different kind of materials and thoroughly characterized them, and then now in this stage, because this is the whole 20 years plus of experience where we have now started exploiting them because we studied deeply these, and there are multiple applications of these, like making materials you can use for targeting, sieving, drug and nucleic acid delivery, biosensing, and whatnot. So in the group, so I come from a big group, around 30 students are there and 5 of academics. So this is a big group. So we are trying different aspects of this materials testing. But I'm going to tell you 2 of these, which I am working currently, and then because of time I cannot tell you all the stories, so I thought I will give you 1 or 2 examples. So these are the glimpse of materials which we have synthesized in our labs, and you can see we have pretty much control over their size and shape, and these materials have been used for multiple different applications. So just the research center theme in my research group is that can we regulate cellular responses through material? Can we design materials in such a way so that we can modulate cell responses? So we have done detailed characterization, developed tools for the materials studies, then cell and animal-based models, and then biomaterials. So I'm not going to go in detail because these all are the links which will be kind of a story. Each of these are a story. So So one example I can give you that it is very easy to, because of the visual. So this is where we wanted to control the migration of the cells. So these are the human endothelial cells which forms our blood vessels. And then the ability of this model. So in this case, cell migration you can reveal, and this is cells are migrating into the artificially created wound. And testing of materials we do routinely to test whether how the cell migration impacts uh, when you add material. So for example, in this case, we could completely halt the same thing. These cells are not moving. So there might be many different stages in between, so that you can study the cellular migration using materials. So response to cells, what is happening. I just wanted to give you a quick overview of this. So the area on which I am working for, say, 15 years or so is the prostate cancer. Why? Because 1 in 5 men develops prostate cancer before the age of 85. And the statistics in Australia is every new case every 2.3 minutes. More men die. I don't know how much you know. I can say more men die of prostate cancer each year than women do of breast cancer. This is a amazing fact, because this is the most common internal cancer in Australian men. And then prostate is a gland which is located somewhere here, and this is a walnut-sized gland. And the prostate cancer is the most common cancer in Australian men. So progression can be either locally advanced, like there is a wait and watch strategy, because they say don't touch pancreas until it is too bad. And then they have a many forms of prostate cancer are incurable. For the practical and testing material, so there were two aspects. One is we selected prostate because it's the ease to do manipulation within the prostate, because the prostate can be reached by external tool. You can inject the materials and then do your research by directly injecting the prostate, right? And then this is not a vital organ after 50 years of age, so prostate is not a vital organ. So you can do research using, that's why the prostate is the model for us. So first area I wanted to discuss is targeted imaging and theranostics. So currently, how cancer or any other disease worldwide is managed, there are 4 different kind of steps. First is somebody has got a suspicion of cancer or a disease. The patient goes to doctor, and doctor says, okay, what you have to do, why don't you go and do some tests? So patient goes to a imaging facility or pathology. There they will run some tests. Usually in case of cancer, they have to go through imaging, where before the test could be performed, this patient has to drink something, right? These are drugs. These drugs are inherently toxic, and imaging, you have to drink around 3/4 liters of the iodine, and which is radioactive. Okay, so this, and then once this is done, the report is sent to the doctor. Doctor goes, and then the second step is prognosis. So whether you have a disease or not, if this disease is there, how bad it is, what is the outcome of the disease, that's called prognosis. And once prognosis is established, then therapy starts. So in case of cancer, these therapies are usually systemic. That means you are given a chemotherapy or a radiotherapy, it's a whole body kind of, and these treatments are inherently quite toxic, and usually if a patient is very weak, doctors cannot give the full dose. And then once therapy is going on, then the patient has to see, doctor wants to see that how the disease is progressing. So again, they will send the patient to the same imaging or diagnostic facility. So this is called response to cancer. So during this 4-step process, the treatments or chemotherapy, these are mostly systemic in nature, and because of those there are lots of side effects. So if you have met a patient who is undergoing a treatment of cancer, you might have seen they have shown lots of secondary side effects. They have lost their hairs, their wrinkles on the skin, liver function tests and kidney functions are not appropriate. So this is how. So what we started thinking actually, how to develop a kind of agent which is a theranostic. Theranostic is the same agent which will used for therapy as well as a diagnostic agent. And that should have a following attributes. For example, first thing, it should be agent itself should not be toxic. So it's a biocompatible in nature. If you can generate in a aqueous medium, that would be great. It should be non-immunogenic. If you have repeating injections, then we know that protein and peptides, they are not good choice because they will release, the body will react against them and they will create antibodies. So that's why humanized proteins and nucleic acid and small molecules may be good approach, macromolecules. Then we started if we can synthesize them at a room temperature, which is not required, but it is good, it's added. If it does not generate any bioproduct and environment friendly and clinician friendly, because if you could convert into the readily injectable kit formulation, it will be great. Then ease of application through current available clinical procedures. So if you want to generate something and then you tell your clinician community to get a big machine to able to analyze, they are not going to help it, because this is a very much distinctive between whatever research which is in university is taking place. It doesn't go to clinic because of this, because this is not clinical friendly. If this is not, they're not going to absorb it. Then cell and tumor specific toxicity. If you can target these selectively to the cells where it has to go, then it's good. And then low cost is not a problem in the developed countries, but it's a problem in, because in developed countries insurance take care, but low cost is a significant. It should be a low cost material or the drug which can be used in developing countries. If it is a multifunctional, that's good. And then less regulatory approval. So because a drug which comes into from bench to bedside, it takes around 6 years of 10 to 12 years of rigorous work. So we just wanted to put some goals, and then we started. So this will look like something. You have one all-in-one kind of approach where you have an agent which can work in, which will work for imaging, diagnostic, and therapy. So we started working on two theranostic agents, which will have a targeting component, which is like when you inject any material into the body on the serum, there are two types of targeting takes place. One is a passive targeting, which is by virtue of 3 S's like size, surface, and their shape. So 3S. And then after this passive targeting, the material will reach for active targeting based on whatever the ligand onto the surface are setting. So these are usually protein, peptide, small molecules, or exploit the surrounding pH and the in-one oxygen tension within the tissue. Then this is the targeting component. Then you should have an imaging component, usually metal oxide, metal organic frameworks, and chemicals, dyes, fluorescent materials, radio labels. They have been used for imaging component. And they are usually centered, which is I'm trying to depict. These are the imaging component. And sometimes if you can convert these into the same chemicals and radiation and biological, these materials are used for therapeutic component. But if you can use, there is a possibility then you can use the same agent which can do imaging as well as therapy. That would be great. So initial studies which I was doing during my PhD, and after PhD we started working on many different kind of ligands while we were working on the central core as well as on the different ligands. So we checked array of ligands which can work, which will not work, and each of them tells a story. I cannot go in detail, but just, so I will take you to only the green tea story. and we got many publications and the patents came out of this. During this exercise, we learned that actually if you make your treatment modality or your drug, you incorporate lots of complexity, you need to do rigorous characterizations, and then this rigorous characterization will lead to more regulatory approval and the issues, and that goes into the more time it eventually. So currently we are having two molecules. One is this is like gold nanoparticle based radiopharmaceutical, which is made from the green tea polyphenols, which is EGCG. I will come into a minute when I'm telling you. This is a like application is a brachytherapy for prostate cancer. Current status is it is in like we are running in a dog trial, client on dogs, because dogs are the only other animals which will have spontaneous prostate cancer. Otherwise, in small animals, you have to inject the cells or tumors, and you have to create. But in dogs, it is spontaneous, and the pathophysiology of the tumor is just like human. So we are working with my old where I did my postdoc, first postdoc in the Missouri University. There we are working and using this material. Second one is a 30 nanometer size material which has a center of iron oxide, which can be imaged through the MRI, and then on the top of this iron oxide there are radioactive copper uh, coating, which is for PET imaging. And then this is a dual contrast imaging agent, which has been we are running a first in human trial for the CAR T cell therapy, where you can load these materials with the cells. There is a regulatory requirement when you take the stem cell therapy, you might have heard, and the T cell therapy, when you inject cells into the body, nobody knows the fate. So regulatory agencies, they don't approve. They are reluctant to approve those kind of therapies. So we had a industrial partner who wanted to create a kind of dual imaging which can tell the fate of these cells when these are injected with the therapeutic engineered cells. They are injected in the body. So we developed this. This is, I cannot speak more, but this is we patented, and then this is in first in human trial. We have loaded this material with 2 human patients, and the results are quite exciting. So now coming back to first story, how it was developed. It's a very simple protocol. If green tea has polyphenols, we have published in 2 papers. You can read because I will be very brief. So you take sodium tetrachloroaurate, which is a gold salt. If you put this polyphenol, polyphenol reads to reduces the gold, and gold comes in AU0 state or AU plus state, and that it precipitates in terms of material nanoparticles. These nanoparticles are being used. So we started EGCG. Why, you should ask why green tea EGCG was used in this case. So we know that EGCG is like it docks onto the receptor which is overexpressed into the prostate cancer, which is a 67 kilodalton proteins on the surface of these proteins are. So when we load these cells with the gold nanoparticles, we can see each cell will take these nanoparticles. Each cancer cell will take these nanoparticles. When this goes into the cells, you can see these cells, these nanoparticles are going, and they are not aggregating within the cellular milieu, and they tend to come out of the membranous suborganelles, right? So they come out of the vacuoles, so they can be used as this tells us that these can be utilized for, because the process called endosomal escape, they can be used as a drug agent. This is another figure where we have seen that because the number of these nanoparticles, cancer cells have taken in huge amount of, and you can see the boundary of these cells, that tells you the cell is still healthy. So themselves, these materials or nanoparticles, they are not toxic even after 24 hours. But and then we went and looked at how these can be employed for the we use blocking, traditional blocking experiment to record that these nanoparticles are going in the cells through a receptor. Although we cannot completely block these receptors, that tells us, okay, there are some other mechanisms also which are taking place for entry. Once these materials can go in a huge amount into a cell, you can test them and using clinical CT scanner. And our radiation physicist tells us that this is a huge contrast you can get. So these are a great imaging agent for CT imaging. And then we wanted to convert them. So if you know the chemistry basic, if you take a gold foil and bombard with neutrons, what is going to happen? This gold foil turns into the radioactive gold. That radioactive gold can be used in conjunction with non-radioactive gold, which is a carrier gold. And if you put with this polyphenol, green tea polyphenol, within 2 minutes, you get this solution, which is like you can fine tune. So this is a kind of a kit component where the idea is the clinicians can buy these solutions, and they can be given this carrier and AGCG, which they can mix, and then they can create the radioactive materials which can be used for brachytherapy. Why gold? Gold has a very interesting half-life, 2.7 days of half-life, and it has been used into the treatment. So it's currently used treatments. And then when we make a radioactive gold, we don't see lots of drastic change in the profile. So these nanoparticles, which are radioactive also, they are quite stable. So then we wanted to, when you inject these into the tumor-bearing mouse intratumorally, you can see these. Normally, if take a traditional radiopharmaceutical, inject into the tumors, the pharmaceutical tend to leach out and they go to distant organs. But in this case, when these nanoparticles were injected, which are radioactive gold nanoparticles, into the mouse, in the tumor, we don't see leaching in up to 24 hours, which is a pretty good sign, because these the most of the drugs will release. So we see more than 90%, 93% dose retains into the tumor, which was a very good sign. Then a single injection of these materials, the gold nanoparticles, led to tumor shrinkage into the mouse. And then the control, which were only treated with saline or EGCG, they had to be sacrificed because the tumor was overgrown, and then we had to sacrifice. But the single injection of the gold nanoparticles radioactive, it led to quite a reduction of the tumor. So these findings have been published in PNAS. And then blood parameters are also like normal as it is in a drug treatment. So these are comparable to other drug treatments. So that summarizes the story that a green tea and AGCG, which is a polyphenol epigallocatechin gallate, that serve as a reducing and coating agent for gold nanoparticles. These coated gold particles are biocompatible and sufficiently stable in biological fluids and serum. The nanoparticles are readily taken up by the cells through a receptor-mediated endocytosis and possibly through another mechanism, which is macropinocytosis. And then they escape. When these are in cells, they will escape from the endosomes. The cellular uptake, cell generates enough contrast, so they can be detected by clinical computed tomography, and then a radioactivity leads to selected killing of the cancer cells. So these serve as a theranostics agent, where same agent can be utilized for imaging as well as therapy and the response to therapy. Because when these tumor could be detected, you could image them through CT same. So because they don't move, That's why one single injection is sufficient for doing so. So this is now we are moving to, after coming here, we are working on to like when can we convert this nanoparticle into a non-radioactive form of therapy, because there is a restriction that you have to isolate the animals for the 21 days, because after half-life, the gold most of the radioactive gold is converted into something else which is non-radioactive. So the idea is if we can use the help of physics, where we can do low energy X-ray radiation, because of the heating and other effects, these cells will be killed. So we are trying to work out on this using non-radioactive form of the materials also. But we have seen that we use Australian synchrotron, where we use a beamline, X-ray beamline to the cells. You can see these are the cells which are growing into the flask. This phase is the cell, and then there is a fine X-ray film which you cannot see over here. But when the X-ray beam is coming perpendicular to this flask, you can see this flask turns into the dark. So if you can see here, hopefully this works, and hopefully you can see. So now the beam is falling onto the cells, and then with time you will see this turns dark, right? You can see this lining is coming. So this is, we are placing the dose onto these cells. And with time, once this irradiation is over, we incubate the cells for 3 weeks and try to see how what is the effect. So these are called colony forming assays, where we have used a control where no nanoparticles were used, and then this is a control which is a non-targeted one, and then this is where you did a targeting. So what we see actually with time and increasing dose, we see a difference like the green tea gold nanoparticles. They have like they are much more potent, although we need to do more experiments in this. But these results are exciting. How are we going actually? So this is one example. I told you the story. Are we going good? Because I think I have eaten up all my time. So let's stop here, and I would take as many as questions possible so that we can make this presentation very interactive.
Sir, due to paucity of time, maybe we might not be able to.
That's why. So we cannot go to secondary.
Yeah. So
I thought
if there is one question which can be asked, I received one question directly from one of the students asking what is the current status about delivery of medicines in nano size.
okay. So so there is a form which is a doxorubicin, a drug. So this is now coming in a formulation which is called Doxil, which is currently in the clinics, like this is prescribed. So another is a drug where the bovine serum albumin has been used in a nanoform. So there are two of the drugs. They are already in the market, but these are on the polymer side, I would say. Still, for the there was a drug based on the imaging, which is taken back on it was based on iron oxide nanoparticles for imaging. So we are still working on that, and many clinical trials are going, but I doubt any metal-based drug has come out so far.
Thank you, sir. Thank you for the answer.
Mm-hmm.
Now, this gives me an opportunity to render vote of thanks to you, sir. Sir, I should admit that you are truly a scientist without borders. This is because of the interdisciplinary work which you have showcased. And thanks, sir, for taking us through a virtual trip of the spectacular world of nanotechnology. Your talk today has definitely inspired the audience with many nascent ideas, I suppose, which now needs self-assembly, as it is very famously told, the bottom-up approach into a novel scientific idea. I thank you very much once again, sir, for your time, and it was really wonderful listening to you.
So before I leave, I just wanted to acknowledge the funding which we get. Actually, our lab is like adequately funding. You know, the funds are never enough for research, but yep, it's a, and this is a team actually. So if you see, most of these are Asians, and the work I could only tell which is done by me and my past students, which are Dedrick Song and Jared both. I wanted to show the gene delivery, but time did not permit. So this work was mostly done by Sunila and Arpita. So I thank all my collaborators and friends here, and thanks for listening. Thanks for your patience.
Thank you very much, sir. Thank you. Amreen, should we move on to the technical session?
Yes, please.
Presentation. Now it's my opportunity to introduce Dr. Alia Begum. I think, ma'am, it is introducing you is very dear to us, and I also feel that it is a homecoming to you whenever you are coming to St.Ann's. Here I briefly present the profile of the chairperson. Dr. Alia Begum is currently working as assistant professor at Department of Chemistry, University College for Women, Osmania University, Hyderabad. She has 20 years of teaching and 14 years of research experience and has published several research articles of national and international repute journals. Her main research interests are in field of synthetic organic chemistry, spectroanalytical characterization, and green chemistry. Ma'am has completed 2 UGC research projects titled Conventional and Green Synthetic Methods and its Microbial Activity, Assay of Mercaptotriazoles, and Synthesis Characterization, Antimicrobial Tumor Activity of Amino Substituted Mercaptotriazoles containing coumarin moiety. Ma'am has organized seminars, conference, workshops, is organizing secretary and convener. She has presented posters and papers in many national and international conferences and symposia. Topics of her interest of teaching are in stereochemistry, functional group chemistry, green chemistry, spectroscopy, photochemistry, synthetic strategies, pericyclic reactions, and reaction mechanism. At present, ma'am is guiding 6 research scholars under her who are pursuing for their PhD. She is a member of several associations of chemistry. I request now Dr. Amreen to present virtual green greetings to ma'am. We welcome you, ma'am.
A very good morning to all of you. Good morning, Amani ma'am, Smita madam, and all the faculty members of St. Anne's Degree College. This is the college where I have done my graduation from 90s, I think 95 to 98 batch. I passed out my graduation from St. Anne's Degree College in 1998. So I'm very happy to come to your platform in a virtual mode. And I thank Dr. Amreen and Mrs. Ramya for introducing me. Yeah.
Thank you, ma'am. Over to you, Amreen.
Thank you, Ramya. Welcome, ma'am. Good morning, all of you. We will start with the technical session. So, ma'am, I request you, I'll be just inviting the participants. So they have been given 5 to 7 minutes of time for the presentation. So once the 5 minutes end, kindly please remind them that they have 2 more minutes remaining and 1 or 2 minutes of the questions which you can ask, or 1 or 2 questions from audience. So we'll start with the first session, has 7 presentations, ma'am. With your permission, please may I invite the first participant, ma'am?
Please go ahead.
So first I would like to invite Dr. Dhruba Jyoti Das, who is a postdoctorate fellow from Department of Biomedical Engineering, National Cheng Kung University, Taiwan. Sir, are you able to unmute yourself?
Yeah.
Yes, please.
Hi, Amreen.
Hi, please carry on, sir.
Okay, so am I audible?
Yes, you're audible, sir. You have to share
Okay.
your screen.
Okay. Yeah. Okay, so a very good morning to all of you, and thank you, Amreen, for the introduction. And it was lovely, and I'm honored to get a chance to talk about my research and also my work. So yeah, so basically, my name is Dhruva Jyoti Das, and I'm a postdoctoral researcher in National Cheng Kung University in a Department of Biomedical Engineering, and I am doing my work under Professor Han Sheng Chuang. So basically what we are doing here is my work is basically on microviscosity based identification of DNA and RNA using rotational diffusometry for pathogen detection. So that is the work we are doing here. Basically, I am doing here. So let me talk a little bit about the background of my work. So DNA viscosity, basically it has an advantage of nucleic acid drug interaction. Also, it can be used for characterization of DNA binding properties. It is used for detecting nucleic acid amplification, and also it is used for cellular response of DNA crowding. But among all of them, detection of nucleic acid amplification is very more important, and also we want to focus more in this because it have the added advantage of pathogen detection and genetic engineering, forensics, and DNA fingerprinting. So in this method, we will be mostly focusing on detecting nucleic acid amplification using rotational diffusometry. So for measuring DNA viscosity, we have some conventional method, which is capillary viscometer, cone and plate viscometers. And the drawbacks with the conventional method, which is it requires large number of samples and there is no downstream application. So this one gives us the idea. So there is a large scope in the development of microvolume viscosity measurement of nucleic acid, which could be done by a low sample volume, shorter time, and micro level detection. Right. So with this motivation, which gives us the motivation of our work, where we want to do the real time detection of pathogenic disease, where we would love to work with COVID-19 and maybe tuberculosis and cholera and this kind of deadly diseases. So this is the overall motivation of my work. So, and after discussing my motivation and the work principle, we have aimed our work to be, first, we want to develop this method. And for developing this method, we have kept our aim for rapid sensitive detection of E. coli using rotational diffusometry. So this is the preliminary thing what we want to do first. We want to work with E. coli first, detect the E. coli and develop our method, and then we want to move for various other pathogen detection. Okay, so far I have talked about the my aim. So let us talk about the methods what we are going to do for in this work. So the method we are going to work is Brownian motion. So Brownian motion is a natural phenomena of small microscopic particles, where according to Stokes-Einstein-Debye equation, Brownian motion is inversely proportional to the viscosity of the solution. So here we are going to use the viscosity change of the solution to find out the change in the Brownian motion of a microscopic particle. Okay, so this is the overall idea of my work, how we are going to do this and how we are going to achieve this work. So first, what we'll do is we'll take our E. coli bacteria and we extract the DNA, genomic DNA from the E. coli, and then we mix our DNA, genomic DNA with PCR master mix, and we amplify the DNA using PCR thermal cycler. So once we amplify our DNA with PCR thermal cycler, obviously there will be hundreds of millions of copies of DNA, and which will be increasing there will be a change in the viscosity of the solution. So once we have our amplified DNA, then we used 1 micrometer of modified genus particle. The size of the particle will be 1 micrometer, and we mix the 1 micrometer genus particle with our amplified DNA, and then we detect this one under a inverted microscope, fluorescent microscope, where we use a small microscopic microchip setup. So this is on the extreme right on the top. This is our microchip setup, where we put a droplet of our particles along with the DNA, and then we detect this one under a inverted fluorescent microscope. And the basic concept will be on the bottom side, we have to enlarged image of our microscopic view. So this is the overall image. So left side one, which shows the particle will be having a higher rotation, because there will be no DNA. When there is no DNA, the particle will be having a higher rotation, and in presence of DNA, the solution viscosity will be higher. So because of that, the particle rotation will be lower, and we can identify this one with a cross correlation, in-house cross correlation algorithm, and we can find out the correlation time along with our measurement. So this is the overall idea of our work. And this is the little bit work progress what we are doing currently. So we have first we have done the bacterial culture and successfully extract the DNA from this bacterial culture. And in the results, first we have done our preliminary studies to check the DNA amplification, and we have chosen 3 different primers for our work, 3 different length of primers for our work. And we have done a couple of thermal cycles where we can see from conventional PCR. So this is a comparison with the conventional PCR and our Brown in motion technique. So with the conventional PCR, with a gel image, we can see the identification of DNA up to 20 cycle in all the cases. So lower than 20 thermal cycles, we don't see much of the amplification. We cannot identify the amplification in gel less than 20 cycles. And also we wanted to check the specificity of our amplified DNA. So we have checked our primers with pneumonia and Staphylococcus aureus, all the 3 primers, and the gel image shows. So specificity of the primer is really good, and we got some specific amplification only. And this is the next data for my Brown in motion rotational diffusimetry experiment. So the extreme left one, first graph is for the correlation time plot with the PCR cycles. So this is the correlation time we have plotted for our amplified DNA with respect to different PCR cycles. So here we can see up to 10 PCR cycles if we compare with the control. So up to 10 PCR cycles, we can see a significant amplification, means we can identify the amplification which is happening. And also the right side image is for the length of DNA. And when we check with our length of DNA,
Dr. Das,
yes, ma'am.
Sorry to interrupt you. Your time is up. Yeah.
Okay. Okay.
Can I ask one question?
Okay.
Can you list out
yes.
Yeah. Can you just list out the ionic liquids which you have used for your DNA studies work? Just what are the ionic liquids you have used? Specific ionic liquids for your work.
Okay. Okay, ma'am. I actually, yeah, actually, that is a, yeah. Okay, so that is actually a different work, and these are the list of ionic liquids which I have used. This is a previous work actually, what I have done in my
Because your title is
graduate
matching
in my doctoral
work.
research.
Yeah. So because your the title which you have given for presentation in the conference is on the ionic liquids, using ionic liquids for
Okay,
your
okay,
extraction. So
okay.
uh,
So
for-
okay. So actually, that is the work which I have done previously in my doctoral research. So yeah, these are the ionic liquids I have used previously for extraction of lanthanides, especially thorium.
okay. So can you just give one application of the ionic liquid uh, which you have used for your work, like
Okay. So
the advantage you
yeah,
observe?
yeah, yeah.
Yeah.
Yeah. So this is my thorium extraction studies with our ionic liquid. So we have used two different ionic liquids. One is the task specific ionic liquid with butyl and NTF2 counter anion, and other one is PF6 counter anion. And this is the extraction of thorium with this ionic liquids, and where we can see with the NTF2 counter anion, we can see a very good extraction compared with PF6 counter anion of ionic liquid. So here we have compared two different ionic liquids, and obviously the first one with the butyl and NTF2 counter anion shows a very good extraction of DNA at 0.01 molar nitric acid solution for thorium. So yeah,
Are they
so this is the,
reusable? Ionic
yes,
liquid
madam.
have used, can we reuse them? Can we use them again after performing extraction? Can we reuse the ionic liquids?
Yeah,
Can
yes, ma'am.
we
Yes,
reuse?
ma'am. Yes, ma'am. We can leach. We can, yeah. So we can leach out the metal which is form the complex with NTF2. We can use EDTA to leach out the metal from the ionic liquid, and then again we can reuse the the ionic liquids for next batch of extraction. But in that, the major drawback will be, it will be the extraction will be little lesser. The efficiency of the extraction will be reduced.
Okay. Okay. Dr. Das, so your time is up. The other
Okay.
participant is waiting.
Okay.
You
Thank you,
just
ma'am.
come to the conclusion at the earliest.
Okay.
1 minute, can you conclude your presentation?
Okay. So if the ionic liquid work, if I want to conclude is so we have prepared different kind of ionic liquids with butyl chain and two different counter anions, which is PF6 and NTF2. And for extraction studies of these ionic liquids uh, has been monitored for thorium, where we have seen transspecific ionic liquids with NTF2 counter anion shows a very good extraction compared to PF6 counter anion. So, and also we can conclude that this ionic liquid has a very good ability for extraction of thorium, and it can be used for extraction of other radioactive elements like uranium, plutonium, and americium as well. So there is a huge scope for in the future for extraction of different other radioactive elements, and for it can be which can be used for our nuclear fuel reprocessing. This kind of ionic liquids.
Okay.
Okay.
Shall we proceed, ma'am?
Yeah. We can
Thank you,
proceed.
Dr. Das.
Okay, thank
On
you.
to the next participant, I would like to invite Dr. V. Jeevana Jyoti, Associate Professor, RBVIR College, Hyderabad. Ma'am, over to you.
Yeah.
Thank you, Amrin. I think I'm audible.
Yes, ma'am.
I'll just share.
Ma'am, we request you to please adhere to the time, 5 to 7 minutes.
Just sharing my screen. Give me 1 minute, ma'am. Stay tuned. My screen is visible, ma'am.
Yes, ma'am. You have to do slide share mode, ma'am.
Okay, ma'am. Good morning, one and all. I'm Dr. Jeevan Jyoti from Reddy Women's College.
Please start.
1 second. I'm sorry, ma'am. I'll just share. Good morning, one and all. I'm Dr. Jeevan Jyoti from Reddy Women's College. Today I'm going to give a brief presentation about my review article on green and sustainable chemistry, a special focus on chemical industries. Chemistry has played a vital role in improving our lifestyle, whether it may be due to medical revolution, that is synthesis of drugs, increased world's food supply, discovery of day-to-day commodities. But all these developments are responsible for environmental pollution. The most important effect is due to release of the chemical byproducts from chemical industries and the release of non-biodegradable agrochemicals, particularly pesticides, into the atmosphere, land, and water bodies. Due to all these, green chemistry has gained a special importance. Objectives of my present review article are: Introduction to green chemistry, driving factors for green chemistry, biomass importance as chemical feedstock, global initiatives, protection laws, and awards for promoting green chemistry, a special focus to chemical industries. Most of the manufactured products involve one or more chemical processes. Chemical industry provides with basic chemicals, materials, and fuels, and has made irreplaceable contributions throughout the history of human civilization. However, it is undeniable that traditional chemical industry is accompanied by a serious waste of resources and heavy pollution. Hence, there is a need for developing a strategy to synthesize day-to-day products by avoiding negative impact on environment. Green chemistry, also known as sustainable chemistry, is an area of chemistry and chemical engineering which is mainly focused on the design of products or process that minimize the use and generation of hazardous substances. Environmental chemistry mainly focuses on the effects of polluting chemicals on nature, whereas green chemistry is focusing on the technological approaches in preventing the pollution and also reducing the non-biodegradable, non-renewable resources. Green chemistry is quite well defined by 12 principles of Anastas and Warner, but these were written 20 years ago and do not fully reflect the modern way of thinking. Additional issues such as toxicology and biodegradability now play an important role in green chemistry as part of a great emphasis on product safety and renewable resources. Sustainable design tries to understand the relation between manufacturing system and ecosystem. Sustainability mainly focuses on the triple bottom line: integration of ecological integrity, societal responsibility, and economic viability. The chemical and allied industries now face a tough challenge. There is a need for the industries to achieve environmental and social acceptability, and also as well as economically viable manufacturing. So there is an exponential growth in chemical legislation, and the very important top chemical legislation is REACH, which is causing major changes in the chemical products supply chain. Sustainable chemical production can only be realized through a reassessment of entire chemical product life cycle, starting from the resources to manufacturing to production, and production to product use, and ultimate fate of the product. Now I would like to mention a few driving factors which are helping in transforming the traditional chemistry to greener chemistry. Legislation is the first driving factor, which I'm going to explain. Legislation and and when most of the customers are pressurizing the chemical industries to strictly follow the safer, greener techniques for the synthesis. After Bhopal gas tragedy, in most of the regions of the world, penalties and restrictions were imposed over the storage of hazardous substances. REACH is the most talked about legislation affecting the chemical products, that is, registration, evaluation, authorization, and restriction of the chemicals. And its highest impact is on the usage of organic solvents like dimethyl formamide, N-methyl pyridine, and dimethyl acetamide. There is another chemical legislation which is mainly focusing on the restriction of the certain chemicals like ROHS, that is, restriction of hazardous substances. Second factor is elemental availability. In addition to the substances which are being restricted because of the strict, which is because of the legislation, there is a chance of having problems with the issues of the elemental supply. Elemental elements which are mainly used by the chemical industry during the manufacturing and the product designing is catalyst. So this problem can be solved by using the renewable carbon, which is a direct replacement, and also by the usage of the metals in a better way. Third factor is renewable resources. Manufacturing of the chemicals mainly depends on the resource dependent. They are resource dependent, and petroleum has dominated the industry as a carbon feedstock. Though now it is a very much greatest demand on the manufacturers to produce biodegradable products by the usage of the biomass. Now biomass has came into picture. So this is the direct replacement of the fossil fuels. So biomass is having a lot of importance as a chemical feedstock. So by 2025, it is estimated that 15% of the 3 billion trillion global chemical markets will be derived from bioresources. The total amount of this available biomass is calculated to be 50 billion tons per year, and out of that 1.3 billion tons per year is from food waste. So food waste is another feedstock which can directly replace the fossil fuels which are used, and before using the food waste, the waste valorization technique we have to implement in order to convert this food waste as the best starting material for synthesizing the chemicals. And there are various major initiatives are taken up at global level, that may be in the form of educational programs, green chemistry centers, networks, institutes. And there are various pollution prevention laws which are being implemented. So to name a few, that is PPA, Pollution Prevention Act, Environmental Pollution Act, and Office of Pollution Prevention Toxins. These acts are mainly enabling the young researchers and the chemical industries to promote green chemistry as one of the important principle whenever they are synthesizing any compound. And some of the awards which are given for green chemistry are listed in this slide, and some of the international organizations which are working to promote green chemistry like IUPAC, ACS, RSC. Now I would like to conclude that green chemistry, which is also known as sustainable chemistry, is to be adopted by every chemical industry in order to convert traditional chemistry to the green chemistry. So now I would like to conclude by saying that, by a quote, someday green chemistry will first be the way chemistry is to be done.
Yeah. Hello, Jeevan Jyoti.
Yes, ma'am.
Yeah. You said that you have written a review article on green and sustainable chemistry.
Yes, ma'am.
Can you just tell me one of the most important aspect pertaining to synthesis, which is the need of the hour to combat pollution, the most important one aspect with which we can sustain the synthetic
Yes,
chemistry methods?
by replacing the organic solvents by the usage of green solvents, and avoiding harmful chemicals, hazardous substances, like renewable resources, natural resources.
Okay. Okay. Thank you, Jeevan Jyoti.
Thank you, Dr. Jyoti. With the permission of Chair, now I would like to invite next participant, Mr. K. Suresh. He's a research scholar in Department of Chemistry, Geetham Deemed University. Over to you, sir.
Am I audible, ma'am?
Yes, sir, you are. Jyoti ma'am, I request you to stop sharing.
Good morning, everyone. My name is Kopala Suresh. I'm a research scholar under the guidance of Dr. M.S. Surendra Babu, Department of Chemistry, School of Science, Geetham, DMITP. Today, I'm going to present a microporous zinc BDCM of aluminum oxide composite for efficient removal of methyl orange, methylene blue, and Congo red dyes from the aqueous solution. Coming to the introduction. Nowadays, the most critical threat to the environment is dyes molecules because of their organic nature and stability in an aqueous medium. Mostly, these organic dyes are releasing from plastic, textile, painting, leather, and paper industries. Different types of different types of methods for removal of dyes, and first one is chemical method, physical method, and biological method. In chemical method, oxidation, electrolysis, ozonation. In physical method, reverse osmosis, filtration, coagulation, flocculation, and absorption. And finally, microorganisms and enzymes coming to biological methods. Absorption is a superior method among these techniques. Absorption of dyes by using metal organic frameworks. And we have prepared zinc BDC MOF and zinc BDC MOF aluminum oxide composite. Composition of MOFs on surface of aluminum oxide has carried the given features: high specific surface area, fine pore structure, excellent chemical stability, and it is low price. It can absorb anionic and cationic dyes also. And this is a synthetic part. We have synthesized a zinc-based aluminum oxide composite MOF by using 1 part solvothermal synthesis. In this typical synthesis, we have taken zinc nitrate mixed with 1 of benzene dicarboxylic acid in a 50% ethanolic aqueous solution and added aluminum oxide in 50 ml Teflon and stainless steel autoclave, and mixture was heated at 110 degrees Celsius for 48 hours. After cooling the reaction, the product was filtered and dried at 100 degrees Celsius for 12 hours. And these are the characterization results we have got. See the XRD structures. The same fakes are avoided, and this is the FTIR, and it shows the morphology. BDC has deposited on the aluminum oxide MOFs. And these are the beta analysis. See the difference between zinc BDC MOF and zinc BDC MOF aluminum oxide composite. Aluminum oxide composite has a low surface area and pore diameter or volume compared to zinc BDC. Zinc BDC MOF has a small surface area, pore diameter, and pore volume. And coming to absorption method, before absorption, we have prepared 30 ppm concentration of methyl orange, methylene blue, and Congo red dyes standard solutions. After preparation of standard solution, we have taken 10 ml of each standard solution and added 10 mg of absorbent. After keep constant under still condition for 90 minutes. After completion of 90 minutes, centrifuge the dye solution and tested for UV absorption test. This is These are the results we have got by using UV visible spectrophotometer. See the absorbance changes absorbance in dyes. Compared to this methyl orange and methyl blue, Congo red was absorbable fastly. It takes only 70 minutes time. And factors affecting dyes. We have tested the absorbance solutions at the various pH ranges from 2 to 10 and taken absorbent doses from 10 mg to 100 mg. We have examined the contact time also. If increase the contact, absorption rate was decreases. Coming to the conclusion, in this work we have prepared a zinc BDC morphine. This zinc BDC material deposited on the surface of mesoporous aluminum oxide, which provide the high performance absorbent to remove cationic and anionic dyes. Zinc BDC of aluminum oxide composites show a better absorption compared to zinc BDC MOF. The investigated material includes the advantages of easy separation and low cost and high efficiency. The obtained MOF composite remains stable and different acid and alkaline solutions and exhibits a good reusability after multiple cycles. All of the results demonstrate that zinc BDC of aluminum oxide composite is a promising absorbent for removal of dyes in wastewater treatment. Thank you. Thank you. Thank you for your attention.
Alia ma'am, you are muted, ma'am. We are not able to hear.
Yeah, my question to you, Mr. Suresh. You have prepared metal organic framework using zinc metal and dye acid, right? So can you
Yes, ma'am.
just
Benzene dicarboxylic acid.
yeah, one difference between a complex and a metal organic framework, or are they same? What is the
The
difference
main
between
difference.
complex and a metal organic framework?
The main difference is, ma'am, MOF has a low surface area and pore diameter, and composite has a large surface area. It can easily absorb molecular dyes.
Yes, good. Okay. All the best, Mr. Suresh.
Thank you, Mr. Suresh. With the permission of Chair, I would like to invite next participant, Dr. Mustaqim Mohammad Abbas. He is a professor in Department of Chemistry, Royal College of Arts, Science and Commerce, Mumbai. Sir, over to you.
Thank you, ma'am. Thank you. Yeah, good morning, gentlemen all. Myself, Dr. Mustaqim Mohammad. Here I am going to present my research work entitled my screen is visible?
Yes, sir. It's visible, but you're very lightly audible, sir.
Yes, now I'm audible, ma'am.
Yes, sir.
Yes. So myself, Dr. Mustaqim Mohammad. Here I am going to present my research work entitled eco-friendly synthesis of some biologically active heterocycles by using nanocatalyst as a green source. So basic aim of my research work was to synthesize a novel biologically active compounds by using some green protocols and the principle of green chemistry, and finally to check out the antibacterial activity of the synthesized compound. So in the abstraction part, I would like to say that heterocyclic compound, as we know, the heterocyclic compounds plays a very important role in medicinal chemistry. And the fused heterocycles like pyran, pyrimidine can be synthesized by a one-pot synthesis by using multi-component reaction. And the reaction used in this research paper is a simple reactant like aromatic aldehydes, beta naphthols, and actylmethylene compound. And the green catalyst used is a new nanoparticle. All the synthesized compound were confirmed by NMR spectroscopy, H1 NMR, IR. Then the reaction was monitored by using thin layer chromatography, and melting point or boiling also confirmed. Now this is about my nanoparticles. It is prepared by two different methods. One is co-precipitation method and second is sol gel method. So it gives nanoparticle size 41.12 and the confirmation was done by XRD. This is my reaction scheme. In this reaction scheme 1, I use beta naphthol, aromatic aldehyde and meldrum's acid as an actylmethylene compound which gives me fused heterocycles containing pyran as a heterocyclic moiety. Similarly, I use benzoyl acetone. Just replacement of meldrum acid by benzoyl acetone gives another derivatives or such type of heterocycles. This is my reaction scheme, and reaction was carried out under in the presence of nanocatalyst as a green catalytic route. This is my methodology. So first, I took equimolar ratio of the reagents. Then I add nickel ferrite as a nanoparticle, then dissolve in a mixture of water alcohol as a solvent source, then reflux it, and reaction was monitored by using TLC technique, and the catalyst finally was separated by using magnetic stirrer, and the product, some of them are liquid, so they are decanting out. This is thin layer chromatography of the compound because this is very important technique for the identification of the synthesized compound. It shows how the compounds are formed. Next is a spectral analysis. All the synthesized compounds, out of which some of them are characterized by using different spectral technique. So IR spectrum shows the presence of carbonyl stretching, zero stretching, and aromatic stretching also observed. Similarly, the NMR of the same compound show presence of CH proton at 4.7 ppm. Similarly, 2-methyl proton at 1.5, 1.4 ppm, and also aromatic proton observed in the range of 6.7 to 8 uh, ppm. Then this is another sample of in which benzoyl acetone is used as active methyl group. Higher spectra confirm the presence of the functional group. Similarly, this is a spectra, NMR spectra, H1 NMR spectra of the same compound. Also gives a clear idea about the CH proton observed at 4.7 ppm. Similarly, CH3 proton of this methyl group at 2.1 ppm, and the aromatic proton at in the range of 6.7 to 8 ppm. So in the result, I would like to say that method is very efficient, gives very good yield. Most of the compound gives more than 60% yield, and yield was compared with HCl also, and as a mineral acid, which is not a green root. So this compound gives very good yield. These are the biological activity of some compounds. This compound show very good biological activity when I compared with and when I performed this biological test, antimicrobial test. So some of the organisms, they are very effective against some gram-negative and gram-positive organisms. So these are the microbiological data. Then these are the microbial strain, and this gives very good zone of inhibition. So which clearly gives idea about the antimicrobial activities. So this heterocycle also plays very important role in multidisciplinary subjects like pharmaceuticals, veterinary. Reference says that this plays very important role in agrochemical also. Polymer additive we can use, dyes and pigment also we can use. Even the food products can be used. In conclusion, I would like to say that we have achieved the synthesis of novel heterocycles. It's a multi-component one-pot reaction, so it follows the green protocol. And the advantage was this method was it's so easy workout. There's no difficulty in isolation of product. Then the use of nanocatalyst find very effective, and it gives obviously the yield is more, so it gives good atom economy, and it can be reused. And all the synthesized compounds show good antimicrobial activities also. And these are the references used for this research work. And in acknowledgement, I would like to say thanks to my director, Professor A. Lakadawala, principal and management of the college for providing necessary facilities. And finally, I would like to say thanks to Green Chemistry especially. Yes, ma'am. Thank you, ma'am.
Dr. Abbas,
Yes, ma'am.
can you please show me the slide which has the structure of the nano catalyst?
Nano catalyst.
Yeah, the structure of the nano catalyst which you have used for your eco-friendly synthesis.
Yes. It is not mentioned in this presentation, but I have this structure, XRD analysis structure. I will not mention in this presentation the structure of this. It's the
So is it a,
molecular formula is here is a molecular formula nickel ferrite particle. Not mention the structure here.
yeah, is it an organic moiety or any inorganic complex you have used as a green catalyst?
It's an inorganic,
Is it an organic?
inorganic moiety is used as a green heterogeneous catalyst.
Okay.
Form of nanoparticle.
Okay.
Yeah.
Thank you, Abbas.
Yeah, thank you, ma'am. Thank you.
Thank you, Dr. Abbas. With the permission of Chair, I would like to invite our next participant, Ms. Tina Renita D'Souza. She is a research scholar from MBJ College of Engineering, Bangalore. Over to you, ma'am. Ms. Tina?
Yes, ma'am, I'm sharing the slide. Oh, ma'am, I'm not able to share the slide.
You are a co-host. think you should be. Do you see a share screen button on your this desktop or laptop you're using?
Ma'am, it's coming. Host disable participant screen sharing.
Tina, I have made you as a co-host, Tina. Nazia Fatima here. So I hope you should be able to share the screen.
Have you logged in with 2 different IDs, Ms. Tina?
Ma'am, could you make Uma Nair as a co-host?
There is another member attendee.
Yes, ma'am. It's a group presentation.
Okay. Can you just spell so that I can find the participant?
Ma'am, Uma Nair.
UMA, that's all right.
Yes, ma'am.
Nazia, only UMA is coming.
Yes. She is the one?
Yes.
Okay.
Yes, ma'am.
Now you please check.
Miss Uma, please unmute yourself. You're the co-host now.
Miss Uma, are you able to unmute yourself?
Yes, ma'am.
Okay.
Should we move on to next participant, ma'am? We'll get back to them in the last. Okay.
Ma'am, we're not able to share the slide. Can we do it after the next participant?
Yes, that's what. Alia, ma'am, can we do that?
Yeah, please go ahead.
With your permission, I would like to invite our next participant, Mr. Darpan V. Bhuse. He is from the Department of Chemistry, Vellore Institute of Technology, Vellore. Sir, over to you.
Hello, is my screen visible?
No, not yet, sir. We can just hear you so far.
Okay. Is it visible?
It's coming now. Yes, now it's visible.
Okay. Okay. Good afternoon, everyone. Myself, Darpan Kumar Bhuse. I am pursuing my research under the guidance of Dr. Pundlik Rambhagat, and my title for the paper presentation is free porphyrin dimer and efficient dye for application in dye-sensitized solar cells. So these are the contents which will be looking: introduction, objective, survey of literature, proposed work, synthesis, characterization, application, and lastly, conclusion. So moving on to the introduction. Now we have heard porphyrin everywhere, that as we know that in hemoglobin we have it's the porphyrin, hemoglobin everyone has heard. So there is porphyrin, which an iron, metal ion is there in that, and in chlorophyll we have applauded too much, and why it has a really good marquee applications in different fields. I have suddenly mentioned those reasons that why porphyrin is used everywhere. The first reason is that it has prodigious ability to harvest light. It can harvest light in the visible region, and it can harvest light in near infrared region. It can excellently perform charge energy transfers. It is 18 pi electron distinct aromatic character, but usually like the fact is that the porphyrin is 22 pi electrons, but from which only 18 pi electrons are delocalized. As I said earlier, it has multidisciplinary applications. there are position, meso position and the beta position, which can be functionalized and it can adapt to the conformation to the local environment. It has excellent optical, photochemical, and catalytical properties as well. Basically, it is a nitrogen ligand, and it has the reaction sites, which I have mentioned earlier, which are meso position and the beta positions. So we'll be looking to the objectives. These are the objectives we have planned. We are planning an engineering of porphyrin dimer, synthesis of porphyrin dimer, its characterization, and lastly, application for its in dye-sensitized solar cell. So what does the survey of literature tells us? That it has immense applications. It is primarily used as sensitizer in dye-sensitized solar cell. It is also applicable in supercapacitors. It is also used in, as a photodynamic drug. It is extensively used as a photocatalyst, and it also has its application in pigments and dyes as well as in the semiconductors. So after dripping the short introduction literature review, we are going to look at the proposed work plan. So we have certain criteria for synthesis of the porphyrin dimer. So the question arises is that why we have selected the dimer in the application of dye-sensitized solar cell. The reason why is that the monomer porphyrin, it can suffer dye aggregation, and dye aggregation has a detrimental effect on the dye-sensitized solar cell efficiency performance. So this, to eliminate the serious effect, we have endeavored in making an porphyrin dimer which can neglect the possibility of aggregation, which in result can deteriorate the performance of the dye-sensitized solar cell. And also, fusing or connecting two porphyrin rings can enhance the ability of porphyrin to harvest light. Now there is now the second important criteria is the incorporation of carboxylic anchoring group. Carboxylic anchoring groups, they are well known to tether on the TiO2 surface, which is obviously a metal oxide surface, which is used to carry the charges, and also it helps to avoid the dye degradation, which is really a big problem. So this is the proposed work plan. Firstly will be synthesizing a metal-free porphyrin dimer. Then nextly will be going for its characterization. Third point is making the photoanodes like the porphyrin TiO2 photoanode, and using iodide carbide as an electrolyte, and at the end we are getting an DSSC cell. So we'll be coming up to the synthesis part. This is the short schematic of the synthesis. I have deliberately mentioned only these two schemes because the synthesis process is much more larger. As you can see, I have used 6 millimole of 2C compound, 1 millimole of 3A compound, and 8 millimole of pyrroles, giving this dye, which is a porphyrin dimer. It has extensive conjugation in it, and the synthesis conditions are mentioned, like the temperature was 80 degrees Celsius, and acid catalyst was used, which is glacial acetic acid, and we have stirred with heating for 40 minutes, getting this porphyrin dimer. Now its characterization. Now, as you can look at the NMR of the compound 3A, the peak are well in agreement with the protons present in the structure. Same with the compound 2. And this is the final NMR of our porphyrin dimer. Now, as you can see that we have got a negative peak at minus 0.125, which is an characteristic peak for the porphyrin, just because porphyrin can show magnetic anisotropy, and magnetic anisotropy, which usually comes because the induced magnetic field run against the flow of the applied magnetic field. These are some more characterizations. Comparative IR of 1B and 3A. The CCl2 disappearance shows the CCl2 disappearance give us the confirmation of CN bond formation. In the figure B, the porphyrin dimer, wherein we can see the COH group, COH group is at 1692 centimeter inverse, and the CN is at 1258, and the C double bond N is at 1395 centimeter inverse. And the porphyrin dimer is thermal 5 degrees Celsius, and we have went for further characterization with the BET analysis, giving us the result that the pore size of it is 0.713 nanometer, surface area is of 500 cc per gram, and pore volume is of 1 cc per gram. Now, porphyrin shows they have a solid band and Q band. So the dye which we have synthesized, it has shown its characteristic nature in this usable spectra. We can see a solid band, 1 and 10 solid band, and 4 Q bands, which is a characteristic of a porphyrin. This is the band gap which we have received through the tau rod method, the band gap which we have received with 1.44 electron volt. And I'll be moving to the application. This is the general schematic of dye-sensitive solar cell. In here, which we can see, we have a conduction band and the valence band. The conduction and band and the valence band is of the titanium dioxide metal oxide semiconductor, and we have HOMO and LUMO for the porphyrin sensitizer dye used. Now, the very general requirement for the dye sensitized solar cell to work is that whenever an photon is incident, the electron from the HOMO goes to the LUMO. Now, the major requirement is that the LUMO should be a touch above the conduction band of the metal oxide semiconductor. So from the metal semiconductor, it will go on the conductive surface, which is fluorine oxide, fluorine doped titanium oxide glass plate, and then the run of the electron flows. This is the IV curve for dye-sensitized solar cell. The open circuit voltage which we have received is 0.610 volts, and the efficiency which we have found is 6.9%. Now we'll be jumping on to the conclusions. So I would like to emphasize on the fact that whenever we were doing our survey of literature, we came across various articles which have showcase the porphyrin dimer giving us the figure efficiencies above 14, 15, 16% of efficiencies. But I would like to say that none of them are metal-free porphyrin dimers. And obviously, our band gap is 1.44 electron volt, which is optimal for dye-sensitized solar cell. The DSSC fabricated gave us the 6.9% efficiency. Again, I have to emphasize on two more facts, is that this 6.9% efficiency has been achieved without any sort of efficiency enhancers, hole scavengers, anti-aggregating agents, and metal doping. The efficiency, I would like to give just 2 or 3 names of the efficiency enhancers, like lithium iodide, tertiary butyl pyridine, guanidinium thiocyanate. Then you have certain amount of ionic liquids and co-adsorbent as well, which is CDCA, HDMA, and DINHOP. This sort of efficiency enhancers can boost your efficiency, can give you double figure efficiency, but those additives, they are detrimental for the long-term performance of the dye-sensitized solar cell. And in here, we have received 6.9% efficiency solely on the base of the raw potential of our dimer and the iodide triiodide electrolyte used. So 6.9% efficiency, I would say it is very much more credible, and I would say this opens a new paradigm which
Sorry to interrupt.
synthesis of the hello.
Sorry to interrupt you. We are falling short of time.
Yeah, okay, ma'am. So I'm just on the verge of getting it in. So 6.9% efficiency has been solely achieved by the raw potential of the porphyrin dimer, and it opens a new paradigm for the synthesis of porphyrin dimer and applicating them in dye-sensitized solar cell, which can give you a greater amount of efficiency without use of any efficiency enhancers and without any need of metal doping. So these are the references which I have followed. I acknowledge RGMS, VIT Vellore, and the management. My sincere thanks to my research guide, Dr. Pundlika Rambabagat. I also thank my Dean ma'am, Registrar sir, HOD sir, VITSF for providing characterization facilities. I'm thankful to my lab colleagues, Smart Material Lab for biosensing and Catalyst for providing lab facilities, teaching and non-teaching staff, SES, VIT Vellore. Thank you.
Yeah. My question to you, Mr. Darpan, is
yes, ma'am.
The structure of the dimer, porphyrin dimer, which you have synthesized in your lab is very complex. Okay.
Yes, ma'am.
And
Yes.
you have used NMR for its characterization.
Yes, ma'am.
And since it has a lot of conjugation, did you record any UV spectra for the dye which you got? UV spectra?
UV spectra for what particular?
For the dye which you have synthesized, because it has
Yes.
extensive conjugation in it.
No, ma'am. We haven't performed its we haven't performed the characterization of fusible.
Okay. And another thing, just based on NMR, you have come to the conclusion of the dimer structure, or any other spectra you have used for its characterization?
Actually, ma'am, in NMR, the most important factor about the porphyrin, or in the broad sense, phthalocyanine, they give you a negative peak, a negative peak. This, what I'm showing is 125 ppm. This is the characteristic peak of any porphyrin or any phthalocyanine, which give us the result that the porphyrin has been formed. And also with the use of this IR characterization, we can confirm the formation of the porphyrin dimer, giving us the respective peaks at positions.
Okay. Thank you, Mr. Darpan.
Okay, ma'am.
Thank you, Mr. Darpan. With the permission of Chair,
Thank you.
I would invite another next participant, Ms. K. Vani from Department of Chemistry, Kakatiya Government College, Warangal. Over to you, ma'am.
Okay, ma'am. Thank you, ma'am. Thank you for inviting me. Good afternoon to all the St.Ann fraternity. Today, I am going to give you a paper presentation over the topic. Shall I share the screen, ma'am?
Yes, ma'am.
It is a review paper that is on the inhibition of xanthine oxidase. It is an enzyme. I would like to present a review paper over this enzyme. Abstract of the paper is the review aims to present the role of xanthine oxidase enzyme that is in uric acid production, and it also its therapeutic effect of xanthine oxidase inhibitors. One of the consequences of xanthine oxidase overproduction results in the gout disease. Generally, it will pain our thumbnail of our leg. It is a chronic rheumatological illness characterized by hyperuricemia, arthritis in association with increased rate of cardiovascular and kidney diseases. These xanthine oxidase inhibitors play a key role in therapy for reducing serum urate levels in patients with gout. Initially, allopurinol, it is a base. It was used as a potent inhibitor to xanthine oxidase enzyme, and febuxostat, it is a non-purine inhibitor of this enzyme. It is a synthesized heterocyclic compound. It is used for the management of hyperuricemia in patients with gout. Allopurinol and its metabolites are structural analogs of both purine and pyrimidine compounds, and thus can affect enzymes in both metabolic pathways, whereas febuxostat is a non-purine inhibitor of only xanthine oxidase enzyme. So in clinical practice, the pharmacological advantages of febuxostat are much over the commonly used allopurinol. So it also included greater serum urate lowering capacity. So this is the abstract of my paper. Next one is xanthine oxidase. It is an enzyme that generates the reactive oxygen species that catalyze the oxidation of hypoxanthine to xanthine, and it further turns the catalysis of xanthine to uric acid. You can see this is the first one is the structure of hypoxanthine. It contains one oxygen atom. It is catalyzed or reacted to form xanthine, or it is converted to xanthine due to the oxidation reaction. That is, the oxidative species will be formed. So, two oxygen atoms are present in the xanthine. You can see here, this is a structure of uric acid. It contains 3 oxygen atoms. So if any liver damage happens, xanthan oxidase is released into blood. So a blood assay for xanthan oxidase is a way to determine if liver was damaged or not. So generally in our lab practices, if liver damage has happened, if they want to check how extent the liver was damaged, that can be determined by checking the xanthan oxidase enzyme level. So because xanthan oxidase is a metabolic pathway for uric acid production, and the xanthan oxidase inhibitors or allopurinol is used in the treatment of the gout. Next, xanthan oxidase is a rare genetic disorder where a lack of xanthan oxidase lead to high concentration of xanthan in blood and can cause serious health problems such as kidney failure. These inhibitors of xanthan oxidase include allopurinol, oxypurinol, and phytic acid. It has been found to be inhibited also by the flavonoids. Among the many known xanthan oxidase inhibitor, febuxostat has been widely used for the treatment of hyperuricemia and gout. This febuxostat, its chemical name is 2,3-cyano-4,2-methyl propoxyphenyl 4-methyl thiazole 5-carboxylic acid. It is reported to have a favorable toxicological profile and high bioavailability, and it is more potent and everlasting or long-lasting hyperuricemic action than allopurinol. Before the advent of this febuxostat, allopurinol was used as a very raw treatment for the patients who are suffering from gout. This febuxostat is the #1 market selling drug for the patients who are suffering with gout. This allopurinol is an orally and parenterally administrable drug for the treatment of gout and preventing the recurrence of kidney stones. So immediately after oral administration, allopurinol is quickly absorbed into the upper digestive tract. It reaches the peak plasma concentration in about 30 minutes after ingestion and has a plasma half-life of about 2 to 3 hours, but a longer plasma half-life of 14 to 30 hours and lower oral bioavailability than its precursor. So the most common documented side effects are gastrointestinal distress and skin rash that can range in severity. Febuxostat is an orally non-purine selective xanthan oxidase inhibitor that can inhibit both the oxidized and the reduced form of XO enzyme, that is xanthan oxidase enzyme, by binding them. So now I am going to propose some of the potent xanthan oxidase inhibitor, which can have a scope for the xanthan oxidase inhibition in the medical field. First one is
Miss
allopurinol
Vani,
or oxypurinol. Hello, ma'am.
sorry to interrupt you. Yeah, we are
Oh,
falling short of time. Yeah, I just
oh,
want to ask you one question, Ms. Vani.
tell me, ma'am.
Topic of your presentation is inhibition of xanthine oxidase, right? So
Yes, ma'am.
generally when we talk about enzyme inhibition, either it has to be reversible or irreversible enzyme inhibition. So according to you, the xanthine oxidase, does it lead to reversible inhibition or irreversible inhibition?
Generally, it must be reversible inhibition. Then only after the completion of the metabolic activity, the drug will be less bind or leaves the enzyme and it will be excreted. If it is irreversible, it will be toxic deposited in the body.
Okay.
It is reversible inhibition, ma'am.
Okay. Thank you, Vani.
Okay, ma'am. Thank you. Thank you to all St.Ann fraternity for giving me this opportunity. Thank you, ma'am. Thank you very much.
Thank you, Miss Vani. With the permission of chair, I again would like to call Miss Tina because you're the last presenter now. Are you ready? Vani ma'am, I request you to stop sharing the screen, please. Miss Tina, please try to share your presentation.
Yes, ma'am. Good afternoon, ma'am. We are from MVJ College of Engineering. My group members are Arshad Naveen, Athira Pradeep, myself, and Uma Nair. And we have done this project guided by Dr. Sayana Varala, Associate Professor, MVJ College of Engineering.
Ms. Uma, we are not able to see your presentation.
Ma'am, we've shared the screen.
Yes, you are allowed to share. You can click on share new screen. Maybe you have opened in a new screen. Do you want us to share for you? If it is, you can send email to us. Immediately we'll share.
Yes, ma'am.
Kindly email to us immediately to the conference email ID. We'll share it. Are you sharing to us through email?
Yeah, ma'am, we're mailing you.
Okay.
Oh, ma'am, meanwhile, can you unmute the participant Arshad Naveen too?
Okay.
Miss Uma, can you please tell the name?
It's unmuted. He's unmuted.
Okay.
Mr. Arshad, are you able to unmute yourself?
Yes, ma'am, I am.
Okay. Do you want us to make him as a co-host?
No, ma'am.
Okay.
Just the audio. Ma'am, we mailed you our presentation.
Okay, just checking. Yes, I received. Just 2 minutes. Is your presentation visible? Ms. Tina?
Yes, ma'am.
Please carry on.
Our group member Arshud Naveen is going to introduce the research rationale.
Yes, please carry on.
Yes, ma'am. Um, the fast-paced industrialization, economic growth, and population growth in developing countries has implicated unpredicted water requirements in cities. Water being the key factor for economic development, as it is used in a variety of sectors. In the last few decades, wastewater production has increased drastically, calling for an effective wastewater treatment. Among various methods for wastewater treatment, coagulation and flocculation is preferred as it is cost effective, reliable, simple, and low energy consuming process. Inorganic and synthetic polymers include alum, ferric chloride, calcium carbonate, aluminum chloride, which were generally used. However, they caused variety of problems to environment and human health, which prompted the need for natural coagulants, which are way more environment friendly and reliable.
Should I move to the next slide? Please let me know.
Yes, ma'am. Yes, please move to the next slide.
The objective is like one of the.
Please let me know.
I'm an objective. I'm objective, ma'am. One of the most popular approaches in to use coagulants like alum, which reduces the repulsive force between the particles, promoting particle collision and flow formation. So, by the recent research, there's a significant disadvantage associated with the use of aluminum salts, like it includes Alzheimer's diseases, which includes the high aluminum residues in treated water, which is actually harmful for the human beings. So we use natural coagulants such as watermelon seeds. It provides benefits such as lower water production cost and had less sludge production and more readily available reagent. So the main objective of the study is to determine the potential of watermelon seeds, which is used as a natural coagulant, and investigate the different characteristics of watermelon. So now we move on to methodology. The first is the preparation of coagulant. So first we collect watermelon seeds and washed off thoroughly. And later on we have done sun dried and ground it into fine powder by using the mortar pestle. So the fine powders are kept in the oven and later on for 1 or 2 hours, and then the moisture contents are removed. So we have done various physical characteristics which are determined by using pH, moisture, surface area, and bulk density. So pH is measured by pH meter, which is calculated around 6.29, and the moisture content is found at around 10.22. And later on, the surface area and bulk density is also determined, which is 0.536 and 10.22 respectively. Next is the preparation of synthetic water. So the synthetic sewage water was prepared using the ground cat food. The cat food which is used here was viscous ocean fish flavor. So the cat food, the 10 grams of cat food was ground, sieved, and dissolved in 1 liter of tap water. The nutritional composition of this cat food comprised of 30% of crude protein, 12% of crude fat, 5% of crude fiber, and 12% of moisture. And this ground cat food, which was dissolved in tap water, was then found using the characteristics parameters such as pH, moisture content, total dissolved solid, total suspended solids, turbidity, and particle size. The moisture content after drying it in the oven was found to be 8.21%. pH was about 6.5. Turbidity in terms of NTU was determined as 18.1. Particle size was about 0.6. Total suspended solids was found to be 920 milligrams per liter, and total dissolved solids was 1020 milligrams per liter.
Next, I'll be talking about the biocoagulation studies. Biocoagulation was carried out by filling 3 beakers with 100 ml of synthetic household water. For coagulation test, 0.3 gram, 0.5 gram, and 1 gram of coagulant was added. Various pH of wastewater were added in this experiment, ranging from 1 to 12. To achieve pH range that was measured, the pH of wastewater was adjusted with the help of using 1 mHCl and 1 mNaOH. The suspension was then stirred for 4 minutes at 100 rpm for fast mixing, followed by 25 minutes at 40 rpm for slow mixing in an orbital shaker. This was done to ensure that flock particles are suspended evenly. After 1 hour of settling, the sample was filtered out, and using portable turbidity meter, turbidity tests were conducted, as well as pH was measured using pH meter, and we noted down the observed temperature. Now we'll move on to result and discussion. For this experiment, pH of wastewater was varied, and it was observed that acidic conditions favored coagulation performance of watermelon seeds in reducing turbidity. It was discovered that pH of 3 is best suitable for this purpose. In the next slide, we can see the tabulated column. As my teammates earlier discussed, the characteristics of coagulant and discussed cat food was tabulated. Next is characteristics of treated water. We can see we took 3 pH range for 3 dosage of coagulants, and due to gradual increment of pH, water became cloudier. Thus, at pH 12, when the wastewater is most alkaline, lowest turbidity reduction was observed.
Hello, Ms. Tina. We are falling short of time.
Yes, ma'am. To conclude, the results obtained from the present study show that the watermelon seeds could be used as a natural coagulant, and the turbidity reduction is observed in all 3 pHs determined, where it was best in the pH 3, which with the dosage of 0.3 grams per liter. And since there is no much change in pH just by the point values, the further studies should be carried out to precisely identify the change in pH to assess the coagulation process, as well as study of the potential watermelon seeds for different quality parameters to make it widely accepted as a natural new coagulant. Thank you, ma'am.
My question to you, Ms. Tina, is why did you specifically use watermelon seeds only? Why not muskmelon or any other seeds for this biocoagulation method or studies?
Ma'am, different coagulants can be used, as you said, but watermelon seeds are widely available and it's much rich in protein, so the coagulation process becomes much easier and eco-friendly.
So are you going to use other seeds also to go for a comparative studies, biocoagulation studies?
Yes, ma'am, we are going to have further studies conducted for citrus fruits as well as the rice husk and compare their coagulation process and the properties.
Thank you, Ms. Tina.
Thank you, ma'am.
Thank you, Ms. Tina. With this, we have come to a successful end of session 1. I would like to acknowledge and express my heartfelt gratitude to Dr. Alia ma'am for firstly taking out the time for this session from her busy schedule and excellently chairing the session with managing the time and all the participants. Thank you so much, ma'am, for your cooperation with us. I would like to thank all the participants and the presenters today. Before we move on, just a small word from our sponsors. I would just like to mention and acknowledge our sponsors for today's event. Union Bank, they are our Pearl partners. They have been like throughout this particular process from 2 months with us, helping us financially, Union Bank, and our additional sponsors and well-wishers, SP Accure Labs and Jolly Scientific Traders. Over to you, Nazia, for further.
Thank you, Dr. Amreen. So to brief up the session 1. It gives me immense pleasure to deliver the vote of thanks to all the dignitaries assembled here. I would also like to thank our sponsors, Union Bank, SP Acure Labs, and Jolly Scientific Traders. I also express my gratitude to Dr. Ravi Shukla sir for igniting the spark among young researchers with his excellent work on greener nanobiotechnology approach or biomedical application. I also thank Dr. Alia ma'am for chairing the session and all the presenters for sharing their part of work done in the field of medicinal and green chemistry. So here we have come to the end of the session 1, day 1 of our e-conference. So before announcing the break, may I request all the participants to fill the feedback by clicking the link posted in the chat box. There are 2 feedback links. One feedback link is exclusively for presenters, which has been already posted by Ms. Deepti, Department of Chemistry, in the Zoom chat box, and feedback link for participants is already posted in the WhatsApp group. So I request the participants, all the attendees to fill the feedback form. So now let's break for lunch and reconnect at 1:55 p.m. Thank you. We are starting at 2 o'clock, right? Next session?
Yes, 2 o'clock sharp.
Okay, thank you, Amreen.
Thank you.
Tell me, Nazi. Hello? Hello? Nazi, you call me? Hello? Nazia, this is Amani. Did you call me? I got a call from you.
Yes, Mama. Can you please mute yourself? Amreen?
Yes, Nazia.
The participants, the presenters list which you have given, I tried searching them, but they are not there.
Okay,
The
I think
presenters, they are yet to join, I think.
I think yet to join, yeah.
Okay.
ఎవరు చేరలే జనాలు.
Amreen, may I request you to keep that slide in sharing, having sponsor's logo?
Okay.
Thank you.
Amrin, can you share that sponsors slide once?
Yes, yes, ma'am. Share it. Just.
Did you get that notification? I once again thank our sponsors, Union Bank of India, SP Acure Labs, Touching Lives Globally, and Jolly Scientific Traders. Thank you. Amrin T. Prabhu Yendrapati, is he a presenter?
Yes.
Okay.
Calling Kalpana, madam. 2 minutes.
Mr. Neetun is a presenter?
Yes.
Okay.
Nazia, madam is trying to join, but she's not able to join, she's saying. Nazia?
Yes, ma'am, I can hear you.
No, madam is trying to join Kalpana, madam, but she's not able to join, she's saying.
Is it? But nobody is in the waiting room also, ma'am.
Or I will give your number to her. I will ask her to call you and speak to you.
Yes, ma'am, sure.
Ma'am, please forward her the link again, ma'am, in WhatsApp.
Yeah, ma'am, better.
Yeah, send the link. Dept, send the link once again. You sent just now? Okay. Okay. Dept sent it already, it seems.
Okay. Because I didn't keep anybody in waiting room, ma'am.
Yeah. Okay, Dept. Huh? Right. Well, time ఎంతండి యుఎస్ఏలో ఉంది ఆవిడ.
Kalpana ma'am has joined.
Joined? Okay, good.
Kalpana ma'am, are you able to unmute yourself?
Yeah, now. Yeah.
Okay. So, Amani ma'am, if you permit, can I start the session too?
Kalpana madam, from my side, welcome once again, and thank you so much for accepting our invite. I am thanking from Chemistry Department, St.Ann College once again, madam.
Thank you. Thank you so much. So I'm sorry for joining late. It's like very early morning here. So yeah, it's around like 4 something. So yeah, early morning 4 a.m. So yeah, I'm so sorry. Yeah, and I'm not able to connect for some time. Yeah, please.
Nazia, you continue.
Okay, ma'am. So, a very good afternoon to one and all. Once again, I, Nazia Fatima, welcome you all to the session 2, day 1 of our e-conference on emerging trends in medicinal and green chemistry, sponsored by Union Bank, Acure Labs, and Jolly Industries. So, I take this opportunity to invite Dr. Amani Das ma'am to introduce our esteemed keynote speaker with the virtual greetings. May I request Dr. Ambreen for the slide? Yeah.
Thank you.
Please accept our token of gratitude, ma'am.
That's so creative. That's so good. Thank you.
So now may I invite Ms. G. Ramya Sri, Assistant Professor, Department of Chemistry, PG, to welcome our keynote speaker.
Good afternoon, everyone. Ma'am, good morning, ma'am. The Honorable Sister Principal and respected Chief Guests, welcome to session 2. It's great pleasure for me to introduce our speaker today, Dr. Kalpana Madgula, SAS Nanotechnologies, LLC, Wilmington, Delaware, USA. Dr. Kalpana Madgula is associated with SAS Nanotechnologies as Research and Development Chemist, Product Development. Ma'am has received her PhD in Chemistry from NIT, Surat, Gujarat. Ma'am received Appreciation Award by Reliance Industries for Catalyst Process Development and one more award from St. Francis College for Women as Faculty Special Recognition Prize. Ma'am worked as Research Associate at Department of Material Science and Engineering, Tulane University, New Orleans, Louisiana, and Department of Chemistry, University of Delaware. Kalpana ma'am has secured research grants as PI and co-PI from University Grant Commission, UGC, Government of India for developing novel polymer composites and nanocomposites. Ma'am in her credit has a patent publication for the studies on stimuli responsive polymer microcapsules. Ma'am in her credit has 13 publications, 7 in process, and 30 plus presentations, 4 invited lectures, and 2 conference organized, and 2 industrial workshops. Ma'am guided 3 PhD students and 10 PG student projects. Ma'am has authored and co-authored multiple publications in the synthesis and characterization of nanomaterials, polymer nanocomposites, and biopolymers that are crucial for catalytic, antimicrobial, and nanomedicine applications. Welcome, ma'am.
Thank you. Would you like me to share the video, the presentation?
Yes, ma'am.
Just a minute. Yeah. Are you allowing me, or how is it? Are you able to see? Hello?
No, ma'am.
No, ma'am.
Yeah, one moment. Okay. I don't see that.
Now you are the co-host, ma'am. You can share the screen.
Yeah, okay. Not able to see the share screen. What about now?
No, ma'am.
I'm not getting share option. I'm not getting the share option. So,
Ma'am, there is a green color icon.
no, I'm not able to see here. Just a minute.
Ma'am, are you using MacBook?
No, not a MacBook. Wait a minute. Share screen now. Yeah.
Okay.
Yeah. How about now? I think now
They're able to see.
So, good afternoon, everyone, and good morning to me, but good afternoon, everyone. Thank you for the opportunity to talk about my work. So I would not be speaking everything about my research because it's too interdisciplinary. So this presentation, basically what presently I am doing, whatever the research I am involved in, and how it is connected to the sustainable technologies or green chemistry. I try to align it. I hope you will be patient enough to tolerate. So how can, like, as Ms. Ramya introduced, right now I am working in a product development in a, you can say, coatings company. This is basically a chemistry company, we could say. So where we are looking at developing a sustainable coatings, we could say. So it's a synonym for green, the word green, sustainability is, where you say that, like, the durability and there is no wastage in the environment. So many other factors will unveil as I go into the presentation. So basically today's topic about my presentation I'm speaking is a smart, advanced and a sustainable green coatings. Since this is a vast area, I could say few parts of the coatings I would be able to present. So what I mean to say smart, advanced and sustainable, because the coatings are there in the industry from long time. So people are looking at making them green or recyclable so that we create a minimum waste. Okay. So these, you must have heard from the morning, there are 12 principles of green chemistry. Similarly, we have 12 principles of green engineering when it comes to the industry level. So when you generate waste in the lab, it would be a little bit like significant in terms of, the material. But if you are generating waste in industry or larger scale, it would be very large. And, it's so difficult to get rid of it. So these are the principles what we follow when we do it in industrially, any laboratory process scaled up to the industry. So it should be like, designed for the commercial afterlife, and it should be renewable rather than depleting, and minimize the material diversity and the less amount of volatile organic compounds. So as I go, I'll be telling you in detail or in specific with respect to the coatings industry, how we use a green aspect of engineering or chemistry. Okay, so green coatings are, what are the green coatings? Green coatings, we call them as environmentally friendly materials. One moment, I'm not able to see the full screen. Yeah. So green coatings, we call them as environmental friendly materials. When do we call them? When we make them with non-solvent bond technology or with low or zero volatile organic compounds. Okay, so what do you mean by the volatile organic compounds? These are the main hazardous chemicals which are present in the coatings, which have to be avoided when you want to call them as environmental friendly. So this is the biggest task throughout globally for all the coating companies, or for all the, I could say in general, any chemistry companies, to reduce them. Okay. Solvent bound coatings. Most of the coatings are very stable, durable when they are made mostly with the solvents or organic solvent based. But organic solvent based, if they are made, they generate most number of like volatile emissions, or because they are highly volatile, they generate emissions of chemicals. Okay, so what they form, if at all we have a vivosis, and you will have a very good coatings or durable coating when you make them with the solvent. Because they are not environmental friendly, slowly all the companies are shifting from solvent bond to water bond. So they are conventional, but there is a need to shift from there. So they reported to form ground level ozone and also the urban smog, resulting into various health hazards. Okay, what is the coatings can be classified? I'm just giving you the general classification because I just want to introduce before I go in detail into my work. So you can either classify them by type, like based on what type of solids or chemicals you are going to use, or based on applications, based on like where they are applied, I could say, like architectural, industrial, automotive, wood, packaging. So everywhere they are there, and based on the geography. Okay, so this is like Latin American European Association. So there is a large number of coatings that would be applied throughout the throughout the world. So based on the type of chemicals what we use, if you dilute them by using the solvent, they are called solvent bond. If you dilute them by using the like water, we call them an water bond. And there are some coatings without the solvent also, so that's what we call it as a solvent free. So water bond and solvent free are considered to be sustainable, whereas the solvent bond are existing because it's so difficult to replace them because of their good quality. But people are slowly moving away from it. Or based on type of coat you apply, whether you apply to the substrate directly, where we call it as a primer, or you apply on the primer, that's what we call it as a top coat for aesthetic purposes, like whenever the surface has to be exposed outside, like for in the case of architectural buildings, or whether you are applying them on the industrial products or on the buildings, or you are applying them to generate some properties, we call them as for some function, we could say. Again is the corrosion, again is the, we could say, environmental degradation, for all that based on properties or function. So they can be classified in all these ways. Okay, so what are the ingredients that make up the coating or that make up any of them? So ingredients, we basically say that like resins, also called a binding polymer, additives, and pigments and fillers, and then solvents. So these are together will make as a coating agents, and they can be applied as a paint or in the form of ink or in the form of a films, hard coatings on woods or on the concrete, we could say, or they can act as an adhesive. So these are the various forms where all these three combine to form as coating agent and can be applied in all of them. What like industrially, which particular part of them are important? So binders, basically the name itself suggests that it bonds the pigment particles to each other and to the substrate. Substrate, I mean to say, it can be a wood, metal, or concrete, or any other surface. Even if I speak earlier, I worked on the you can say the coatings or films which are applied on the medical devices. I could say catheters or any of the medical device equipment which is used. So what are the like some of the important resins which are consumed in large amounts? I have just like highlighted them. The rest of them are also available, but industrially, what I work are what most of them are like alkyd resin, acrylic resin, epoxy resin, and polyurethanes. So these are the form of polymers which can be used as a binders. And you have one more component, we call them an additives. So which can be added in small proportions, sometimes to improve properties or sometimes to create a gloss. Gloss, we call them as shine, or sometimes to adjust the rheology or flowability of a coating. Like sometimes we apply the paint in the form of a spray, sometimes we brush them, sometimes we apply as a thin film. So based on what type of application you are looking at or thickness of the coating you are looking at, the liquid or flowability of the paint will change. And the most important part is solvent, which can combine all of them. So here the solvent, either it can be dissolved, that is the pigments are dissolved in the solvent to make them in the proper way of coating, or sometimes the binder won't be dissolved but can be dispersed. For example, if you're using an organic solvent, you are going to dissolve pigment, additives, and fillers into the organic solvent, and you make like a uniform dispersion and apply. When in the case of a waterborne coatings, you cannot dissolve all of them in the water. Instead, we call them as disperse. So in the waterborne solvents, the particle size of pigment, the particle size of additives, and the binder and water dispersion, we call them. Because this is a polymer, and if you are using a solvent as a water, the dispersion between the binder and the we can say water is very, very crucial. So here our chemistry plays a major part where the uniform dispersion you would be making, which will have a single property, then each of them are showing their own individual property. So it should have a uniform property. No component should express itself. So here are the, either you call them as a pigments, or sometimes this pigment can also add up the color. When you are using it as a top coat, it can add up color. And pigments are also can be inorganic or organic type. And we call them as fillers. Sometimes these fillers will add up the strength, will act as a barrier property, will also act as an extender, we call them as. Then what does the composition look like? What all we mix in this to make it as a proper composition or proper paint? Normally the paint formulations never be called as a green. But can we make them green? Yes. People are looking, the companies or organizations are looking to make them sustainable and green. The main two components, what we are looking like, I'm not talking about other things because a large number of research is going on each of this component. But my talk will be mainly focused on replacing the solvent, like organic solvent, that's what to reduce volatile organic carbon emissions or VOC emissions, with water, like see 58%, almost like close to 58% we are able to replace this formulation. and also large amount of pigment. These are the two large components which will form among the formulations. Rest of them are very small components. And how do you make them green? You use water instead of solvent, and critical pigment volume concentrations we call, that is more solid content should be there as compared to any of them, because binding polymer, again, it's a part of polymer which you cannot replace easily, because you need a binder which can bind all of them together. And dispersant you need, because water cannot get mixed with polymer if you don't have a dispersant. Defoamer, when you actually grind and mix, there should not be any foam generated. Foam is nothing but an air, isn't it? So when air is generated, it won't allow particles to stick together and won't form a uniform dispersion. Rheology, you need a flowability based on the type of application, and coalescing agent to blend all of them. And if at all you require any additional additives for functional purposes. So if you're using this paint for some function, antimicrobial, anti-corrosive, anti-fouling, so whatever function we are looking at. So what is the critical pigment volume concentration? This is very important. If a particular formulation will allow more and more pigment that can be dispersed in the composition, that would be a good formulation, we call it as. So how do they apply? So paint application would be normally two parts, like you select a substrate and, select a substrate, and substrate would have an interface, we call it as a solid gaseous. And another is, the second is like you apply a liquid paint film, and there is an interface between solid and liquid, and you dry or cure, we call it as. A curing can be air dried or heat dried or radiation cured. So there can be so many parts. So you will have a uniform coating onto a solid substrate. This is the way we apply based on its interface properties. So how do you make it like environmentally protection? If you look at, there are three aspects we can make. Use high solid paints, because reduce the volatile matter and increase the high content of non-volatile matter. That is, reduce volatile organic compounds, because they are going to cause high, high depositions in air, and it's not environmentally acceptable. And replace the solvents, organic solvents, whatever you use, either dispersant, binding polymer, to some extent, use the polymer resin, which is, made from the biomaterial. And another is, don't try to reduce the solvent-free systems. That is, either use them two component, that is one component to make it liquid, another component to dry, or cure them with radiation, use powder coatings. Okay, why is it so important? Like most of the organizations are transitioning from high volatile to water based, where you have the component of organic constituents is almost like 200 grams per liter, and mid level, and to the low or zero level. So this is the transition happening. Somewhere we are existing over here, mid level to low. So people have moved away from high volatile, because strict regulations are implied now. So why is it important to replace solvent bar? They are doing extremely well, but the consumption is 33 million tons globally, the architectural coatings. So this much is employed or used globally. Then, the VOC created by the usage of this much will be significant. So that's the reason every country is enforcing to replace them. Okay, so why they have to replace? Major focus is on sustainability. So two aspects it would be majorly looked at, because it is the major portion which present in the pigment formulation. So waterborne, and also the pigments are additives. So here, pigments are additives, the two components I have mixed. That means that use the pigments which can also act as functional additives. So you are making two components into one component, because you are using the pigment and also which is a functional additive. So that is what like our technology is about. So I'm taking one example of where the pigment has been used as a functional additive to protect a corrosion, like to protect the metal from the corrosion. So that's what like we have like patented publication on that. Okay, so pigments are additives. If you use with enhanced functionalities, you can replace one of the component in the formulation, that is reduce one component, and also you use such pigments which are sustainable or which can be easily recyclable, which can be easily degraded. So why is it important? Like why you need to have a corrosion inhibiting additive? So, the cost of corrosion is so much where not only the material will be degraded, there will be a lot of energy wasted, economic and human life will be wasted. So if you replace at least one of the substrate, that is like if you protect most of the substrate, you will save replacing at least 1 ton of steel, like, which contributes to the material making or which contributes to the construction. So what is a corrosion? It's an electrochemical process which is induced by the corrosive ions which are present in the environment. Now, our coating or formulation should have the barrier which prevent corrosive ions to reach the substrate metal. So substrate metal should be protected from the environment of corrosive ions. So how are the corrosive ions are produced? So corrosive ions are produced when material reacts with the environment around it. So the environment consisting of moisture, oxygen, or any harsh environment. I could say when the material is in operation in an industrial process, there can be a temperature change, there can be a chemicals present. So what type of coatings normally applied? So depending on the type of coating, the operating environment or the protection time. Some of the construction, if you say bridges and all, they would be harsh environment, but they require protection longer. So it depends on the lifetime of the sample. For example, steel will be durable for longer time compared to the simple iron, or aluminum will also be a durable. So based on the lifetime of the sample, you apply a particular method and you apply a particular coating application. So why is it necessary to have? So the demand for the anti-corrosive coatings, so it looks like we see them and we don't know their value. That way it is like the coatings, what they apply, it is almost costing them 22 billion US dollars. There are two aspects. One is an environmental aspects, because the right now people are using the coatings which heavy metal pigments such as zinc compounds, lead compounds. When they scratch or when they remove the coating, these zinc compounds are introduced into the water streams. So which will cause health, safety and environmental, because you are using so much of coating. I showed you the 33 million 33 million tons of coatings you have been using, and when they are removed or cleaned, you see how much it will be going into the water stream. One is environmental, the second is now the regulation. So this is called one of the regulatory body, we call them as a REACH. So any coating composition, any formulation you develop, it has to be accepted by these two organizations. So one will be the European Union and another will be the United States. So this has to be followed globally. That is REACH we call, and another is Toxic Substance Controlled Act. So this is Toxic Substance Controlled Act is in general for these two, for all the chemicals, irrespective of coatings. It may be a coating, it may be any industry. All the chemical industries, pharmaceutical industries should follow these two regulations. If these are not accepted, you are not supposed to develop any scale-up process. Okay, so what are the concerns like, okay, we have to meet these two. What are the concerns? So concerns are whenever there is a scratch or mechanical damage, the substrate will be exposed and the coating will become ineffective. So as long as there is no scratch, there is no environmental effect, coating is fine and it goes on. But once there is a damage, the whole component needs to be replaced, which will cause huge amount of loss for an industry or for any organization. So that, and another one is solvent based. So you replace them and again you are making a new coating and you are consuming more amount of VOCs, generating, I could say, more amount of VOCs. So now they are looking for the intelligent and smart coatings. What do you mean by the intelligent and smart coatings? They respond automatically for the mechanical and chemical damage, and they reproduce with their original properties. That is, they can have a good adhesion to the substrate and integrity. And another is sustainable. They should be environmentally acceptable. They should be easily scalable and blended into the formulation. So whenever you introduce one component in the formulation, you are not supposed to change all other components, because everything to redevelop, it's not a feasible. So that's the reason that whenever you introduce any component or any change, make it sustainable, make it easily blendable into the existing formulations or existing technologies. So these are the few examples I have taken. People have developed for various areas, that is, which can be triggered by biological, or which can be triggered by the chemical, or by light, or by thermal. So I told you the one which can respond to mechanical or chemical damage. So a tumor, for example, there was a tumor, and when you trigger it with the light, the material can respond and can treat the tumor and also can regenerate the skin. So similarly, the same mechanism has been applied where you are developing functional additives. So I can divide in general the strategies which has been gone into the developing such smart coatings. Either you use the additives directly into the polymer matrix, or you use the additive, a hybrid, I could say, hybrid system where hybrid oxide and the functional additive has been mixed into the coatings. And the third one is coatings containing micro or nano capsules. Okay, I'll go quickly with the things. I don't know how much time I have. So I'll go quickly with all this, and I'll go to the exactly what the work we have done. So what is a polymer coating? So polymer coatings, we introduce like self-repair properties of polymers. We introduce a corrosion inhibiting compounds into the polymer matrix directly. So these are the compounds which can, consider the function of inhibiting the corrosion onto the substrate. And the properties, the matrix and also the compound should have. The matrix should have oxidation properties. It should be able to swell and cross-link, and no high temperatures are required. So the smart functionality is based on either the additive what we mix, or can be additive and polymer matrix blend. And there are some combinations already existing, fluoropolymers, polyelectrolytes, and you have very famous RGC, ruthenium Grubbs catalyst. So this also have a company that is based on the ruthenium Grubbs catalyst. They have acquired the funding and they started the company. RGC is in Los Angeles in California. But the problem is that this is expensive and unstable, but very, very durable and very successful application is this one. And another is based on conducting polymers and also based on the epoxy materials. So these are the type of materials which have been used, a polymeric materials, where functional additives have been mixed into these polymeric materials and made into polymeric coatings. So few examples, for example, a Nippon company, we call it as, it has produced a super absorbent polymer in the form of a capsules. So whenever there is a scratch, the particles of super absorbent polymer will be released into the layer, into the scratch. That is, they form like a glue and close the scratch. So once the scratch is closed, it can reduce oxygen diffusion from the solution, from the sap layer, and protect the underlying, we can say, substrate. So this is one technology they have used this company. And another was based on, we could say, the epoxy resin. Why I am saying what is the specialty of epoxy resin is, this is already existing in the market. Epoxy resin is being produced regularly. So it's so easy to introduce it into the already existing resin, the functional component. So what are the functional components I have introduced? Malamides. So here the protection is into two forms. The protection at the crack, where malamide will combine with the thiol to form a cross-linking network and close this crack. That's near the crack. And the protection even is there in the bulk, where the same malamide, that is malamide you have mixed into the malamide component has been mixed into the, as a functional additive into the epoxy resin. So maleimide will react in the bulk with the residual amine and also will close if at all is there any the leakage in the coating, or is there any oxygen diffusion through the pore in the coating. So basically you want to provide a barrier where oxygen or moisture will not reach the substrate. So the barrier here is this two combination, and barrier here is thiol and maleimide combination. So another one is a JIP mechanism, we call it as, where the isocyanate groups, whenever the coating is exposed to the environment, the isocyanate groups will absorb the moisture and will form like a JIP-like mechanism. They form into a stable urea cross-link. So these are the isocyanate groups combining with the water and forming a JIP, or closing the leakage, whatever has been happened in the coating. So what are the hybrid oxide and composite coating? Some of them applied. So this is the recent research I am telling, 2018 onwards, because there are so many that have been developed earlier, but I'm not talking about them. This is the only recent one. So here also the phosphate groups, which are, acted as a surface functional groups onto the graphene oxide. So there is a graphene oxide on either side, and ends of the graphene oxide have been functionalized with the phosphate groups, and these phosphate groups form a stable protective film where they block the entry of oxygen or chloride ions. That is, most of the chloride ions are being replaced by the phosphate group, and phosphate groups will form a protective film or passivating film on the surface, so that they would not allow any more ions or any more corrosive ions enter into the substrate. So this is another work people have done. And what is the speciality of coatings containing microcapsules? So as compared to the other two, this is the one which is now taking into effect. What is the speciality? So this is here nano capsules or microcapsules. That is, you make a capsule which is of the size of nano or micro. That is, it can be a micrometer or nanometer, and into the capsule you are introducing a functional additive or whatever your function you are looking. Either you introduce a foulant, antifoulant, you introduce a corrosion inhibitor, even for the drug delivery you introduce a drug, and you want this capsule to release the component where and when needed. Not like, that is, you are enclosing them in the capsule, you don't want them to release if there is no need. So here the idea behind this is the capsule will only release when it is triggered by light, heat, or pressure, or it can be a change in the some environment, change in the phase, or change in the pH. So you have the additives, functional additives enclosed in these capsules, and they are blended into the matrix and are coated onto the substrate. They are there, but they are not released. So where you require only small amount of functional additive, that is, you are reducing the quantity, and another is you are not wasting it. When there is no scratch, when there is no damage, you don't need them to be released. When there is damage, you want them to release. So that's why the durability of these additives would be increased. So what is the speciality? Like, is it easy to make such nano or macro capsule shells? There are a number of factors we take into the when you are making or when you are tailoring the caps, a number of factors we have to take into the attention. So thickness, morphology and sizes, chemical composition, and also how do you blend them into the paint or polymer matrix? If you blend them, are they going to change their characteristics? Yeah, everything, every aspect of the property you have to look at. So controlled permeability, that is, you introduce them, but they are not released at the specific time. So that's why you have to make them permeable. Whenever they are triggered, they have to release the additive. And another is adequate strength. Till the time is needed, they need to retain their strength, and they should have a long shelf life. With the time, they should not react with any of the components. And excellent bonding to the host material, and non-toxic, preferably sustainable. Otherwise, whatever may be the technology, it's not acceptable. What can be a shell materials? What type of shells we can make it? So we need to use only these type of polymer materials which are acceptable and existing. You cannot just use or make any of the shells. And you can have like most of the materials which can be enclosed. These are enclosed materials or encapsulants. So these are the materials we normally use. Like for example, polyurethane, polyaniline, polypyrrole, cellulose, chitosan, and also some of the clays, that is inorganic base, also have been used as a shell materials. I'll take one of the each of them as one example. So how do you choose a preparation method to make a capsule? So there are a number of methods available. We call them as emulsification. That's what the technique we have used in our, in our work. Emulsification, where you form a monomer droplet, that is a droplet of monomer. For example, I could say aniline droplets. The aniline droplets are being stabilized in a solvent, or water, I could say. And these droplets are being emulsified by the, emulsifying agent or oppositely charged agent, chemical agent, and they form like a shell around a droplet. This one will be hollow. That is, there is nothing inside. It's a hollow capsule. Or you can make like a layer by layer assembly, where actually the permeable hollow capsule, this is not permeable. You need to make it permeable, or you can need to keep like it is hollow, but you need to introduce something. You have to make it like a pore, or a, open, partially open. And this one is two oppositely charged polymers have been aggregated into conservates. And then interface separation we call, that is, polymer is dissolved into a core material with volatile solvent, and after that the solvent will be either precipitated or will be evaporated, I could say. So how do you release? Okay, you have encapsulated. After that, how do you release? A release can be a chemical method, a release can be a physical method. So chemical method, we remove the depolymerization, or we remove the cross links. And physical method can be a pressure. I told you, like with any of the trigger, light or thermomechanical. So these are some of the existing things where already people have tried to introduce, like triethanolamine, they have introduced, and they use it for all these substrates. Okay, so where are the applications? So these are the existing ones already people have used. People have used like titanium nanotubes, where they have loaded with epoxy monomer, that is monomer droplets have been formed with, which has been enclosed with this DDA dodicylamine into the epoxy monomer, and they are being introduced into the titanium nanotubes. And these titanium nanotubes are introduced into the coating matrix to release them slowly. And also, there are like polyurea formaldehyde capsules or urea formaldehyde capsules, which are made like capsules and also introduced into the matrix for the slow release. And another is, this is very, very much famous and very much looked forward, because the reason is halosite nanotubes. They are available, you know, they are available naturally like that in the form of a tube. So which are considered to be environmentally acceptable. So these are, in the, is there anything wrong? Excuse me. Okay, so here people have applied halosite nanotubes inside a coating, where mercaptobenzothiazole has been introduced into the nanotubes, and a polyelectrolyte shell has been used to close the nanoshell.
Someone is having queries, question.
Okay, so if you have after introduce, how do you evaluate whether the coating is intact or whether the shells have been leaked? So we use like techniques like electrochemical impedance spectroscopy, which will we give the quick check where the coating substrate is being introduced into the matrix, and we run the plots, apply the frequency and measure the resistance of the coating. So for example, I'll explain with this graph. As you immerse the coated substrate into, into the solution for number of hours. Like with increase in the immersion time, there is increase in the healing property. So the coating has to be healed or unaffected when you increase the immersion time of the coating. So this is a quick check what we do, the integrity of the coating. And another we use is scanning vibrating electrode technique. We call the specimen is taken here, and with the probe, we look at the whole area of the specimen. That is, this probe would be vibrating, where we introduce oscillations, and we'll see whether the coating would be intact or it would be removed in the presence of an oscillation. So this looks like, the graph looks like you don't have any functional additive introduced into the coating. Then the control look like there would be a current flowing or current density increasing, because there would be corrosive ions which are present here. And when I have a coating intact, which is modified with capsules, so here the capsules, whatever the particles, whatever the capsules are encapsulated with methyl benzothiazole as a functional additive. So there is no current density. That is, there are no corrosive ions which are introduced, because this is acting as a protective in the coating. So the two graphs can be compared after we apply the coating with respect to the control. And another one we use is scanning electron chemical microscopy. We call electrochemical microscopy, where you want to have a small sample size, I could say, where sweat won't provide the change in the chemistry or chemical changes. So here bare or non-coated surface, and here the coated surface. The coated surface will give you the less current, and uncoated surface will give you the more current, because of the presence of ions which are affecting the substrate. So this is what industrially people will use. We call it as for long-term applications, a salt spray test, which we use like a ASTM method. That is, you coat a specimen with the required coating and keep a scratch and immerse it in either in, in the salt solution, which has been made according to the ASTM method, or you spray the salt. Okay, and look whether the coating is intact or got spoiled. So this particular test is carried out for at least 500 hours, 1000 hours. That is, if you want a durability, like for construction applications, you don't want to replace any of the material within a year or 2 years. You want them to 10 to 15 years. You want to see it long-term application. So that's what every industrial coating looks like, 500 hours and 1000 hours. So this is the acceptable test industrially. So whether you perform all the other three, you have to perform this to show that your coating is intact. And another, so these are the things we have looked at. So what are we looking at? We are looking at long-term and robust protection, reproducible and scalable, and to be able to blend into the existing formulations, and it should be sustainable, inexpensive, and meet the regulations. So this is what we are looking at. And here onwards, like I'm showing my work. So glimpse about my research. So these are various companies I have worked at, and various research areas I have worked at. So during my PhD, I have worked at organic and nitrogen sulfur containing compounds or inhibitors, and also I have worked at inorganic organic hybrid precursors during the R&D at Material Reliance Technology Center. Francis also, I had some polymer nanocomposites. Tulane University have worked on biocompatible iron oxide nanoparticles, and presently this is the stimular responsive polymer macro nanocapsules. This is actually the patented work. I could only show the pictures because it's like pending approval. So that's the reason. So these are the areas what I'm looking at. I don't have much time, if I believe. Ramya? Ramya?
Yes, ma'am. 5
Yeah.
minutes.
How much time I have?
5 minutes, ma'am.
Yeah. I'll just wind up. So this technology, as I told you, the smart microcapsule technology, we have one, a National Science Foundation, the grant, and also another seed grant. We call it as from University of Pennsylvania. That is this idea like encapsulating a functional additive into the capsule has won like a lot of awards. These are the 3 awards. As a company we have one, it's not an individual one, I could say. And one of the technologies has been patented and also been accepted by the Oxo Nobel uh, paint company and won in the global competition. This particular idea of smart microcapsules as functional additives. And another, we have like 2 patent applications. One is like a composite application, another is a macrocapsule application. So how it looks like, our technology, since I am not telling like what is the polymer and what we are enclosing, I will only tell the mechanism. Okay, so these are like smart polymer macrocapsules. There is like, a hole, I could say, the emulsifying technique we make. So they are hollow inside with partial open. So we make the shells with partially open and we fill it with the chemicals. And these chemicals, once they are filled, they were only released into the environment by one of these triggers. That is, we allow the shell material to be triggered by one of them. So that's why the shell material is very, very important, because you produce a hollow shells where any of the chemicals can be introduced. For example, I can introduce a drug material where drug can be released into the body, triggered by, I could say, sustained released by, triggered by pH change, triggered by some enzyme like that. So that's why this particular technology is open to any of the sustained release techniques. So that's why this is, we call it as a platform technology. So first we produced a shells which are partially open, and these are all the applications we are looking at. This is already commercialized. We are like scaling up and commercialized, and we are looking like, the chemicals can be a followants, chemicals can be a lubricants, a pesticides can be introduced where they can be triggered whenever they are needed to for the agriculture purposes. This is also, we have one more project where we are working with the agriculture department. And also you have some of the specialty chemicals and also the drugs. So this is like, the normally the capsules are introduced onto the primer. I could say this is a substrate where we have a coating in which the capsules have been introduced and also applied onto the substrate or onto the metal, for example. So whenever there is a damage or whenever there is a scratch, okay, so then the corrosive ions will enter onto the substrate and will spoil or degrade the substrate. But when the scratch is made, one of the capsule would be broken. So once the capsule is broken, the functional additive will come into the picture and will not allow the corrosive ions to reach the substrate. So that's why once there is a scratch, there is a capsule releasing and sealing the surface. So capsules will be everywhere, uniformly distributed into the primer or uniformly distributed into this coating. So that's why the capsule is only releasing its additive only there is a scratch. So when there is no scratch, when there is no damage, then there is no release. So that's why you are protecting and using only the few amount of chemicals, because you don't need to release them when there is no scratch. So here we are saving one, cost of the chemical. You can say durability of the chemical. It can be for longer time, because when there is no scratch, there is no need of release. So that way we are saving on cost and also on the environmental, protection happening. So it can be like, applied as a primer to the substrate. Normally we apply not for the aesthetic purpose. Sometimes we require a top coat for like, aesthetic purpose or look good purpose. And this is a market we are looking at. Like, large amount of market is still unopened. These are the companies which are already existing, and lot of them small companies are still developing here. And this is another technology where the pigment particle with pores has been polymerized on its surface. That is, a pigment particle will have a pores on its surface, and in the pores we polymerize and make like a composite. Okay, so this is also like a composite pigment particles are introduced into the, onto the surface and into the coating. And this also has the same application. and this is the second technology, and how they work. So they can form like an ionic barrier. They won't allow metal and toxic ions. And also when the barrier fails, even the barrier fails, due to the difference in electrochemical potential, they can activate, microcapsules can be activated and can then release the additives. So since the scale up and commercialization is in progress with these two technologies, so these are the references what I have taken, and I think I'm on time. Yeah. Yeah. So this is like disclaimer. Hope I have. Sorry. Shall I
Yes,
stop?
ma'am.
Yeah. Hope I am
Yes, ma'am.
on time. Okay. So I
Okay.
think that this is different from what you have seen mostly. Okay. Thank you. Thank you.
Thank you, ma'am. I would like to propose vote of thanks for today's webinar. As we express our gratitude, we must never forget that the highest appreciation is not to utter the words, but to live by them. I would like to thank our distinguished speaker, Dr. Kalpana ma'am, for sharing your views on paint resins, polymeric coatings, and enlightening us with your knowledge. We are all inspired by your great words, ma'am. Thank you, ma'am.
Thank you.
If any participant is having any questions, you can post in the chat box. If anyone is having any doubts or questions, you can ask.
Is that like all of them students or?
Narjis ma'am, Narjis ma'am, you have any question?
Yeah.
Amrin ma'am, you can unmute Narjis ma'am.
Yes, ma'am. Narjis ma'am, please unmute yourself.
Good morning, ma'am. Like one question I have, what is the time period required for the whole the process, whatever the technology that is a platform technology you're applying, ma'am?
You mean to say time period to prepare, or what is that exactly?
Like whatever, the flow chart you have shown, no, ma'am. So, from starting to the end, what is the time required for the completion of the total process?
Like for capsule formation and encapsulation, you mean to say? Yeah, it would differ on the quantity. As we know, any chemical process, like whether I'm making it as a gram level, kilogram level, or that. So basically, it would be a 3-hour process, laboratory process, I could say. If you are making it in gram level, and I could say this is at room temperature. So that's where the environmental thing comes. You don't require energy. So it can be run at ambient temperature. And in water, I could say. In water, no organic solvent, in water and room temperature. And 3 hours, yes.
Okay, thank you, ma'am. And ma'am, one more I have, ma'am, how to choose which compounds are appropriate for a particular filling or coating and all, how to get an idea,
Yeah, that's a really good question. So normally, that's what, like in the beginning, I told you, whatever we choose, it should be part of one of the components. That is, it should be easily blendable in the existing formulation. So normally we take ideas from one of the binder resins. Okay. So I told you different components: binders, solvents, additives, and the dispersants. So normally, whatever people have done research, so the resins which are used as a binders or which are used as a polymeric materials, from them, if you are able to make capsules, one of the resins, like I could say polyurethanes, urea formaldehyde resins, and if you can make a blend of resin with additive, like if I'm looking at a anti-fouling, or if I'm looking like antimicrobial. So your antimicrobial additive should be easily blendable or easily encapsulated into the resin. The blending between polymer resin and additive should be very good. If there is not very good or compatible, you cannot encapsulate it. Okay, because they exist like a two components. You want them into one component, a shell with encapsulate, a shell with functional additive. The shell is polymer and functional additive can be any antimicrobial compound. So the blendability is very, very important. So most of the time existing resins people will take, because after you make a capsule, it has to go into the coating. So you know that the resin is already proven. So that's why if you choose any other resin, you have to prove it from the resin. So existing resin, if you take, it is proven, so it's easy to blend. So any existing resin of any formulation you are looking at, look at the formulation where you want to go, look at the resin, that's it.
Thank you, ma'am. Amrin ma'am, you can unmute Amani ma'am. Amani ma'am has one doubt.
Amani ma'am, your co-host ma'am, you can unmute yourself.
Good afternoon, Kalpana madam. I have a basic doubt. This coating will depend on two factors. That's what I'm thinking, whether it is right or not. Just let me know from your side. So the coating, what we are going to do the coating, that material should be, it depends on the where we are going to use that one. Or we are going to give the coating on what material we are going to. These two are the factors will influence the coating usage, madam.
Exactly, ma'am. Exactly. Yes. So it depends on the substrate where you are going to apply exactly.
Yeah.
And in what environment? What environment? Yes, exactly. Yes. That's the basic difference. Yes, ma'am.
Ma'am, this is Ramya. I have a question. Can I go ahead?
Yes, ma'am. Yes.
Ma'am, my question might be slightly off track, but this question has been lingering in my mind quite some time. If you visit this very old temples or Buddhist cave temples, you see beautifully painted walls which have withstood hundreds of years of nature's fury, whatever it is. So, is there any work which has been taken forward in finding out what plant pigments have been used, and why can't we because we know that all of them will be either it will be from a mineral source or it will be from a plant dye. So why are not people trying on in that way and use the same as a greener pigment? Because in many of the paints, pigments are either heavy metals or organic dyes, and we know the kind of damage dye is doing, and people are already working on dye degradation 24/7.
Exactly. Yeah, that's really good question. Very good question. See, it's happening, like the coatings are very stable. Like for example, we have like number of research papers on that. People have worked like, old porcelain components where they told that gold and silver nanoparticles have been introduced into the composition. Even some people have proved carbon nanotubes have introduced. They don't know that is nano. Later on, once the microscopy is discovered, we know that the particles are nano in size. But without knowing, earlier our ancestors have already worked on nano. Okay, so that's what, like if you look at in recent papers, gold and silver carbon nanotubes have been used. And another is with respect to the metallic components. Like during my research, also I have done Qutub Minar, you must have known in New Delhi. So there is a metal pillar, Ashoka pillar we call it as, that is withstanding corrosion. Like it is not corroding at all. They are not painting it. Okay, it is withstanding from somewhere, I think Krishna Devaraya dynasty something. Somewhere they have introduced the pillar and still nobody is doing anything to it. That has been thought to be, there is like internally, they have used some soldering technique or, the metal forging technique where the metal has been treated like, with hammer. They have done the metal with hammer so many times that it has made in such a way that heat treatment, what we do now, earlier they have generated the heat by actually doing a hammering work. And also the pillar, wherever it has been kept, at the bottom, we call it as a cathodic protection in electrochemistry. The pillar bottom has been connected to 4 more metals which are sacrificing to protect the pillar. That is, the 4 metals are getting electrochemically degraded and helping the pillar to stay in its place. So every protection, whatever happening from our history, has a different mechanism to follow. That's what I want to say. Some coatings, unknowingly, they have been made, naturally stable because unknowingly they did it and it proven. Some have been degraded, and people are using natural pigments. Earlier, what is happening, if you look at, our ancestors have not much pollution they have done, and with the time pollution is increasing. So whatever coatings earlier has been used are becoming ineffective now in the existing conditions. And when you are using sustainable pigments or sustainable materials in a large scale, it is not cost effective. Okay, so that's why industries and if I want to use an environmentally protective pigment or dye, to scale it up, I have to consume more amount of I could say green resources. Okay, if I have to make 1 gram scale, fine, but if I have to make 1 kilogram scale and 50 kilogram scale, I'll be consuming natural resources a lot. Okay, so that's why cost effective is one thing. Are you actually protecting the environment by spoiling the environment? That is also one effect. So that's why the technology which is acceptable in both way is definitely coming up. In Europe, people are doing a lot because European regulations are very stringent. So people are looking at the sustainable. Yes, they are looking at that. For example, like antimicrobial, I'm hearing turmeric being used, neem is being used, but like very few have gone into scale up level or industrial level. So we look at natural polymers, biomaterials, like what we are working, which we want to scale it up. Our work is biomaterial. So which is not like synthetically made, but biomaterial. It's not like, what do you call, environmental harm it will cause. Yeah.
Thank you, ma'am. Thank you for such a nice explanation. Over to Amreen, ma'am. Ma'am, can I introduce next? Okay. Good afternoon once again. I am very much impressed to introduce our chairperson for today's session, Dr. Naresh Kumar Katari. Dr. N.K. Katari is an assistant professor in the Department of Chemistry, Geetham, deemed to be University, Hyderabad, India. Sir has obtained University Gold Medal in MSc and PhD from JNTU, Hyderabad. Sir has 11 plus years experience in the areas of synthetic organic chemistry, material science, and other multidisciplinary field, and has more than 10 years of teaching experience in various reputed universities. Sir has been awarded several awards such as Excellence Research Award and Best Teacher Award, etc., and guided 2 PhD scholars. Sir has been thoroughly engaged in the research work, guiding 11 of his scholars who have enrolled at Geetham, JNTU, Vignan University, and JNTU, Hyderabad. Sir has published 67 papers in various journals. Sir has secured 30 plus articles published in international and national journals in several areas. Now I ask Dr. Amreen ma'am to welcome Sir with green greetings. Sir, welcome you, sir.
Thank you so much, madam. It's my pleasure to be part of our international conference on medicinal chemistry and green chemistry. Thank you so much for your good introduction. Thank you. Then shall I proceed with the session, madam?
Yes, sir. Please.
Yeah. Please, you can start the session, madam, with the first project.
Yes, sir. So with the permission of the chair, I would like to invite our first presenter today of the second session, Dr. Gunvanti Negi. She is Assistant Professor from Department of Chemistry, Royal College Arts, Science and Commerce, Mumbai. Ma'am, over to you. Nazia, can you please make ma'am as co-host?
Yes, I have unmuted now.
Yes.
I'll just share the screen.
Yes, ma'am.
Madam, the screen sharing is disabled. It's showing.
We are cross-checking, ma'am. One second.
Yeah.
Now it will come, ma'am.
Now you please check, ma'am.
Yeah.
Ma'am, we request you to please stick to the time, 5 to 7 minutes, ma'am. And I request the chair to please follow the time schedule. Thank you.
A very good afternoon to one and all. My topic of presentation today is the analytical application of ion selective electrodes in the determination
Sorry
of...
to interfere, ma'am. We are not able to see your presentation.
You're not able to see?
No, ma'am.
But I can get the indication that you are screen sharing.
Yes, ma'am, but the PPT has not been opened. It's just in the files, ma'am.
Okay, I'll just try again.
Yes, ma'am.
Madam, can you see now?
Yes, ma'am.
Okay. Good afternoon to one and all. My topic of presentation today is the analytical application of ion selective electrodes in the determination of strontium metal ion in clinical samples. Starting with the introduction for this topic. Now, we all know that strontium is an alkaline earth metal, and its compound has a number of applications. When we talk about the determination of strontium, we basically focus on a number of sophisticated instruments like ICP-AES and like neutron activation analysis, etc. Now, these methods are quite expensive for routine quality analysis, and therefore simple rapid methods are required, which would be cost effective for routine analysis. And since the first development of strontium selective electrodes in 1975, only a few strontium ion selective electrodes have been reported. Basically, our work was to develop a strontium ion selective electrode. Due to size compatibility of strontium with 18 crown membered crown ethers, we had used ditergbutyl cyclohexano 18 crown 6 as the ionophore, which was selective to strontium ions. And after the electrode was developed, we had used it in the determination of strontium ions in clinical samples of blood and chicken bones. Basically, strontium is an important element and has chemical similarity with calcium. Also, a compound of strontium called strontium ranelate is used in the treatment of osteoporosis. And the method which we have used is a simple potentiometric method. The research study is divided into different parts. The first one is to fabricate the strontium ion selective electrode. After that, we had prepared the clinical samples and we have done the potential measurements. For the quantitative aspect, calibration curve method was used. Whatever results were obtained, they were compared with the ICP-AES method. Coming to the fabrication of the selective electrode for strontium. First, a mixture of different ingredients was made.
The composition was 33% PVC, which is polyvinyl chloride. Basically, this is added to provide mechanical stability to the membrane. Then we had added a plasticizer, orthonitrophenyl octyl ether, 55%, and 6% of sodium tetraphenyl borate, which acts as an anion excluder. And to increase the conductivity and surface characteristics, nanotubes were also added. The composition was 2%. And the selective crown ether for strontium, we had used 4% di-tertiary butyl cyclohexano 18 crown 6. And these ingredients were added in the solvent tetrahydrofuran. After this, the components were sonicated and coated on a graphite electrode. Then the electrodes were dried and conditioned in strontium solution for 48 hours. Coming to the preparation of clinical samples, the blood samples were prepared by using standard procedures in which concentrated acid like nitric acid was used and hydrogen peroxide was also added. Then the solution was boiled, cooled, and spiked with known volumes of strontium standard. In a similar manner, the second sample of chicken bones were prepared. The bone samples were dried under sunlight for 3 days and then they were incinerated and converted into ash. 1 gram of the ash sample was taken and treated with nitric acid, and then they were spiked with known volumes of strontium standards. So this is how we had prepared our sample solutions, followed by standard solutions for the calibration curve. This is the potential measurements which were made. So simple potentiometer was used, and on the left hand side you can see the calibration curve for the strontium ions using the developed electrode. The cell assembly which we had used is shown. So you can see a saturated calomel electrode was used as a reference electrode, and our indicator electrode was a developed graphite electrode. Using the above cell assembly, we had got the Nernstian slope of 29.1 plus minus 0.3, and we had used this electrode for determination of strontium in the clinical samples. Now coming to the calibration curve method, in which different standard solutions were prepared, and we had recorded the potential of the standard solutions, and the equation of the line which we got was 29.84 log C plus 206. Now using this equation, we had calculated the concentration of strontium in the different samples. After that, they were compared with the ICP-AES analysis. Coming to the conclusion part, the blood samples which we had taken in this study did not show the presence of strontium, which indicated that the person whose sample were collected were not having any exposure to strontium ions. And the bone samples which were analyzed indicated the presence of strontium in the range of 0.37 plus minus 0.14 ppm. The results which we had obtained by using the developed electrode were found to be comparable with the ICP-AES method, and thus the ion selective electrodes provided us with a simple and a cost-effective method for the determination of strontium ions. These were certain references which we had used for the work. Thank you.
Yeah, thank you, Dr. Kunvati, madam. It's really a
Thank
good
you.
presentation. Is there, can you go for first slide, madam, if you don't mind? I have only one question.
Yes, sir.
Yeah, title, please.
Yes, sir.
Yeah, only simple question, madam, that is relevant to your topic only. What is the rationality to determine the strontium in clinical samples than any other ion? Is there any reason, madam?
See, sir, strontium basically is basically in blood. It is having chemical similarity with calcium. So calcium is very important as far as bone is concerned. As far as in our blood, calcium levels are to be normal. So strontium similarity it has with calcium. So sometimes what happens is because of the presence of strontium in excess level, see there is a level of around 50 nanograms per ml that should be present, beyond which it is very harmful. So that is why in clinical samples the strontium level should be monitored.
How did you get these clinical samples, madam? From where you are going to get?
Yes, from the blood bank, I have taken the blood samples.
Okay, but this strontium is one of the radioactive element in our nuclear
Yes,
waste.
sir.
There is
Yes.
it's my opinion only. We can recover that strontium for various medical applications.
Yes, sir. Actually, we had done in one pharmaceutical applications of strontium ranulate, which is coming in Glenmark. It is coming as Stronate sachet. So in that, we had monitored the level of strontium percentage recovery. We had used, we have determined using the developed electrode. So that
And
work we
what
had.
is the analytical application here?
Pardon, sir?
What is the analytical application used for this determination of strontium? Potentiometric method, no?
Yes, sir. Potentiometric method we have used. It's a simple potentiometer we have used.
Yeah. Thank you, madam. Is there any other questions, please? Any questions from audience? Yeah. Thank you, madam. I would like to invite
Thank you,
a
sir.
participant.
Thank you, sir. Thank you, ma'am. Next, I would like to invite Mr. Nitun Maju Varghese. He's a research scholar from MVJ College Engineering, Bangalore. Over to you, sir.
Respected sir, good afternoon. So I would like to share the screen now.
Yes, sir. Please go ahead.
Ma'am, is the screen visible?
Yes, sir.
Yes, sir. Hello, ma'am. Hello.
Yes, sir. Please go ahead. We can see.
Sorry for the delay, ma'am. I'm a student from MVJ College of Engineering, and we have been doing a research on the immobilization of biomass. So it involves the basic adsorption studies. Now, I would like to start with the research rationale. In recent years, the adsorption has gained a considerable attention due to its high selectivity, operational simplicity, and economic efficiency. The native biomass has disadvantages such as uneven particle size, low density, poor mechanical strength, rigidity under variable conditions. The commercial exploitation of the biomass-based biosorbent system is seriously hindered due to the following technical challenges, like the instability of biosorbent in harsh chemical environment, the insufficient data about the quality of the treated water for useful applications such as agriculture, and the disposal or possible application of the sorbent system after saturation with heavy metals. Now, the main objective of our research was to develop a rigid biosorbent from rice husk via immobilization method for direct industrial wastewater treatment. Now, the main method of our project was we first took the rice husk from a local vendor, and we then took a certain amount of it and washed it with distilled water 3 to 4 times. We then sun dried it for a period of 24 hours and later placed it in a hot air oven for 2 to 4 hours. Once the dried husk was obtained, the way we determined it was dried by taking the weight of the rice husk at regular intervals like 30 minutes, and when the weight was constant, we knew that the certain amount of moisture has been liberated. Now this dried husk, we kept it in a mud pot and sealed the lid the mud pot using a now, as you can see, we kept it in a mud pot and sealed it using MC, and further we then kept it in a muffle furnace at 500 degrees centigrade. Later after 1 hour we removed it from the muffle furnace and kept it at room temperature to cool it, and we ground it using mechanical method to form the biochar. Then we prepared a sodium alginate solution by adding 2 grams of sodium alginate and dissolving it in 100 ml of hot distilled water at 60 degrees centigrade. We then added the biochar to the sodium alginate mixture and kept it on a magnetic stirrer for continuous mixing. Then we prepared a calcium chloride solution for the preservation of the biochar. Once the calcium chloride solution was prepared, we took a syringe and with the help of a syringe, we extruded the biochar and sodium alginate mixture into the calcium chloride solution for preservation. Later, we prepared a standard stock solution of methylene blue. We used 0.005 grams of methylene blue and distilled it and added 100 ml of distilled water to it to prepare a 50 ppm solution. Now, using this 50 ppm solution, we prepared different concentration solutions such as 20%, 30%, 50% methylene blue solutions. To this we added after this we fixed a certain amount of S/L ratio, and we added 2 grams of immobilized biomass to each of these samples and kept it in an orbital shaker at 140 rpms for 4 hours. At the end of 4 hours, we could see the difference in the color and then the results were obtained like this. The immobilization method followed here resulted in immobilized rice husk biochar heads of size around 3 mm. So these are the beads that we obtained, and these are preserved in calcium chloride solution. We determined some properties of the biochar such as pH and bulk density too. Now you can this determines the absorption studies that we had determined. Now you can see the color difference between the initial concentrated solution and once the adsorption had occurred. Now moving on to the conclusion part. The immobilization method followed here resulted in immobilized rice husk biochar beads of size around 3 mm. The adsorption efficiency of immobilized biochar was investigated using a synthesized methylene blue dye solution. Now, based on this, we can use this method in industrial applications for using it for wastewater treatment. Thank you.
Good, Mr. Nitin. Any questions from our gallery? Dr. Amirin, madam?
Yes, sir.
Can we proceed with Q&A session?
Yes, sir. We can proceed.
Any questions, please?
Those who are unable to unmute can post the question in chat box, please.
From my side, there are no questions, madam.
Okay, sir. Then we'll proceed to the next participant, sir, with your permission.
Sure, madam.
Next, I would like to invite Ms. Mansi Gandhi She is a research scholar from Vellore Institute of Technology, Vellore, Tamil Nadu. Ms. Mansi, over to you.
Hello, good afternoon, everybody. Ma'am, are you able to see the presentation?
Yes.
Okay, good afternoon. I am Mansi Gandhi and going to present on the topic biomedical aspects of nanoparticles. Firstly, I belong from VIT or Vellore Institute of Technology.
Sorry to interrupt, Mansi. Can you please make it presentation mode?
Okay, I'm sorry. Is it now?
Thank you. Yes.
Sorry. And I'm from Vellore Institute of Technology. It is a beautiful campus and located in Tamil Nadu, Vellore. Uh, Firstly, I will start with the basic of nano level as my topic initiates with the biomedical aspects of nanoparticles, wherein the first is what is exactly a nano level or a nanoparticle. Any nanoscale level of material is considered as a nanoparticle, or a billionth or a meter is a nanoscale, wherein the most common example is a single strand DNA having a diameter of 2.5 nanometer. These substances have unique properties which are size dependent. It was initiated by Richard Feynman, and the whole exploitation of nanoparticles today, in 2020, has reached a market of around $1.5 trillion worldwide. Wherein, can we see it? Yes, we can, but only the bulk. The nanocarbons, the multi-wall carbon nanotube, or other carbon allotropes, we can see it by the naked eyes, but the exact structure, shape, and morphology can't be visible. So we have to use microscopes like optical microscopes, SEM, and so on. Where did it all started? The next question which comes to our mind. Initially in 1916, as I say, Robert Feynman initiated with a St.Ann as a code, there is a plenty of room at the bottom. Later in 1985, Croto discovered fullerene, which further pushed this work. Finally, after 2 decades, we have a number of different nanoparticles ranging from different size, shape, structure, properties like nanotubes, nanorods, nanocluster, nanocoating, quantum dot, nanocomposite, and the list go on, wherein they have enhancement in their properties. They are either produced via physical method, that is top-down approach, or via chemical, biological, or microfluid method via bottom-up approach. The top-down approach involves the bulk being into a powder and further exfoliating into different layers, finally into nanoparticle. While the bottom-up approach involves a precursor, which connects together via nuclear formation, and finally nanoparticles are being formed. After the formation of nanoparticle, the most important part is the characterization. How do we know what exactly are the structure, the composition, the surface morphological features? Therefore, different characterization like PL spectroscopy, that is photoluminescence spectroscopy, which is used in order to understand the light and the photon, that simulate the emission of photons from any matter, followed by TEM to understand the surface characteristics, XRD to understand the crystal structure, composition, size, AFM, atomic force microscope in order to understand the 3D shape, NMR or the nuclear magnetic resonance in order to understand the purity, conformation, structure, MOSBER, FTIR, XPS, and ICP-MS, and there are other very different techniques which are quite crucial to understand the interaction and basic properties about the nanoparticles. After the properties, their applications comes into the picture. They have numerous amount of application. The first basic application which we feel in our day-to-day life is a sensor or a glucose sensor, wherein different glucose, as in where the protein or the glucose is detected by an electrochemical based sensor which is on a nanoscale level. A small chip is used to do all this work. Further, the tissue engineering, commercial systems like food wrap or what we call as the intelligent wrapping systems are one of the best examples of nanotechnology, wherein the smart wrappings using gold and silver nanoparticles are being developed. Further, the wall paints are incorporated with nanosilica, which has water repellency properties, followed by tin oxide that absorbs the UV visible wavelength. They even band-aid, the medical systems are using nanoparticles, which increases the surface area and even has antibacterial properties, specifically for nanoparticles of silver. Later, medicines are also coming to the picture, wherein they have hydrophobic or hydrophilic properties of the pharmaceutical drugs, which have the feasibility and better stability. Later, it has been extended to optical coding, cell manipulation, and so on. Even the skin creams which are used in our day-to-day life has tin oxide nanoparticles. Apart from nanoparticles, they have huge number of limitations, as in they are very expensive, They are equally toxic. That is, they have a capability of evil potential. Moreover, Moreover, when we are exposed to these nanoparticles on a day-to-day basis, they can lead to mass poisoning over time. That is, inhalation of nanoparticles can lead to settlement inside the body. It can even be weaponized like a nanobio warfare or a smart bullet. This has even caused a huge exploitation of the people in the traditional ways of manufacturing and industrial sector. Even new agriculture concepts are being come up with the use of nanoparticles. Further, there was a concept known as grey grow scenario in which they have thought that a self-replicating nanopod might consume everything and make copies. Even the lack of knowledge of nanoparticles can create a huge havoc in the industry. Apart from this, challenges are very common, which we have observed in here. Very basic scientific challenges are being there, starting from sample composition, chemical status, reproducibility, nanosymbols, and so on and so forth. Further, the current scenario wherein they have actually used in our day-to-day life, the first example is Adventus, wherein they have used for the drug delivery approach, wherein polymeric nanoparticles are engineered in order to carry out the anti-tumor drug across the blood-brain barrier. Even a company known as BASF have come up with a toothpaste having hydroxyapatite nanoparticles, which improves the dental surfaces because of hydroxyapatite in it. Further, there's one more industry, the Science and Technology, which has come up with an aeroside filter having TiO2 nanoparticles. They are used to destroy the airborne pathogen. Finally, I come up with future possibilities where expansion of nanomaterials into drug delivery and other pharmaceutical therapeutic pipelines can help in improving the efficacy. Moreover, the practice to revisit the status of suboptimal and biologically active molecular entities have made it through the conventional approaches and even entering the field of tissue engineering. Moreover, these nanoparticles have their endless properties, which can be a boon or a bane equally and can even lead to toxicity. Hence, for a more smarter, advanced, and yet safer system that can trounce various daunting challenges associated with its application. These are a few of the references that have been used, and thank you.
Yeah, really, it's a good presentation, Miss Mansi Gandhi.
Thank you. Thank you, sir.
Any questions, please?
Any questions from the audience? Sir, any questions from your side, sir?
Yeah, Mansi, can you go back, please?
Yes, sir. Slide. Yes, sir.
Show me your second slide.
Second.
Yeah. See, there are two approaches no, to prepare our nanoparticles. One is top-down and another one is bottom approach.
Yes.
Which approach you adopted?
Pardon, sir?
There are two approaches no, to prepare the nanoparticles. Which
Which
approach
are
you have chosen?
both of the approaches are being chosen in our day-to-day life. Particularly what we top down is more complex, but even like for our setup we can use electrochemistry to actually decrease silver
Okay. Then
solution.
may I know the name of your nanoparticle?
Silver,
What is the
common
name of
silver
your?
we can take and we can make nanoparticles out of the silver solution.
But what is the novelty in this? Because these are well-known approaches, no?
Yes,
Where
sir.
is your novelty?
Novelty, the work which I am considering is actually, it's a review work wherein all the from the initiation of the starting of nanoparticles have been kept on with how it has come into our living existence using its applications, and finally what are the disadvantages or limitations of using it.
May I know your designation in VIT?
I'm a research scholar.
Research scholar.
Yes.
Who is your supervisor?
My supervisor is Rajagopalan, sir.
Oh, Rajgopal. Okay. Thank you much. Yeah. But presentation is good. It's too good. Dr. Amrin, madam.
Yes, sir.
Please proceed with next participant.
Next participant, I would like to invite Mr. Taraka Prabhu. He is a research scholar in Department of Analytical and Structural Chemistry, CSIR IICT Hyderabad. Sir, over to you.
Madam, can you see?
Yes, sir. Please make it presentation mode, sir.
Yeah.
Yes.
Good afternoon, one and all. I thank the whole St.Ann's College management for giving me this opportunity to present my work, creative design of a facile ternary composite ZNO, ZNS, NIS heterostructured for efficient photocatalytic hydrogen production. I am being guided by Dr. B. Sridhar, Senior Principal Scientist in the Department of Analytical and Structural Chemistry, CSIR, IICT. And our team has been doing, our group has been working on this photocatalytic activity, mainly on the degradation of pollutants, water splitting into hydrogen evolution, photocatalytic CO2 reduction to methanol. All these three works have been done by our group. And we know the photocatalysis can be, this approach, photocatalysis, we can do things like degradation of organic dyes, splitting of water, organic transformations, CO2 reduction to valuable added products, all that. Except the organic transformations, the other three we are doing. And before doing any of these works, the important thing that we need to know why we are using these semiconducting materials, because they need to harvest a big amount of solar energy, because it's a photocatalysis, and therefore it can be used to convert chemical energy for resolving the global energy demands and environmental issue. The photo-induced hydrogen evolution reaction from water by appropriating the band gap structures. We need to appropriate because it's a reduction potential, and the oxidation potential needs to be matched in such a way that our desired products are achieved. In obtaining this, there are four things, that factors we need to look into. Any catalyst that we make, photocatalyst that we make, it has to have a wide spectrum of light absorption. And once the light is absorbed, the photons, they have to be charge transport and separation needs to be done quite effectively. And during this separation and the transport, we need to reduce the recombination of those photo-induced electrons, photo-induced ions, electrons and holes. And the surface properties of the charge transfer also need to look into. Giving the basic understanding what exactly this hydrogen production by water splitting. We know the water and some energy is needed, and for that, because of that energy, the water splits into hydrogen and oxygen. The solar light is here used as an energy required to break the water molecule. This catalyst photo activation in this photo generated holes in the any material that we take, it will have a valency band and a conduction band, and it continues to remain. Once the photons fall on that, the electrons in the ground state go to, from the valence band goes to the conduction band, and there they get from there the activity begins. And we know if you take any semiconducting material, the light is induced, electrons in the conduction band and holes in the valency band are formed, and we need to know the reduction. Especially we need to concentrate on the reduction potential here. The reduction potential for hydrogen is at 0 electron volt, and the oxidation potential is 1.23. Any material that we are making, must have these appropriate engineering of the band gaps. Therefore, then only we will be able to successfully split the water. And now, therefore, any material that we have, the conduction band must be above the reduction potential, and the valency band needs to be higher than the oxidation potential. And the materials that we have synthesized are zinc nanorods. We took zinc nitrate and mixed with HMT hexamethylamine, and you can see that, and mixed in the water, just allowed aging for 24 hours at 85 degrees. The nanorods are formed, and followed by, we allowed it Na2S for sulfonization, and again we did mixed in the water particular amount, and at 60 degrees, and the sulfur is decorated on the nanorods, And we took the zinc nanorods with the sulfur, ZNS. And again, we put the nickel nitrate and thiourea we have mixed and put at heating of in autoclave method around for 12 hours at 175 degrees. Then we collected after 12 hours, we collected the material, centrifuged it, and we have dried it overnight, and ZNO, ZNS, NIS is formed, and we have varied the percentage of loading of or decoration of nickel sulfide on the zinc rod, zinc ZNO at the rate ZNS rods. And we have taken the XRD, we have taken for all the materials, for all the three pristine of NIS, ZNO. Therefore, in the XRD, we came to know the structural composition that exactly it was matching with the JCPDS numbers of the nickel sulfide, ZNO, ZNS, all the XRD clearly shows. And coming back to UV spectroscopy, as we said, harvesting of light, we know the NIS has a large amount of harvesting. When we mixed with or when we decorated with ZNO, ZNS, started to absorb more and more light, and the band gaps are also being calculated, and we have done the surface DET to know the pore size of it, surface of it, and we know the composite material ZNO, ZNS, NIS 2% at 0.02 molar concentration. It is showing that surface area is quite high for it. And we have done for the XPS where we will know the oxidation states of each element, and it proved that what our material is well synthesized, and its oxidation states are intact, and it can be very much seen in the XPS. And coming to the morphology, as we were going on telling that we have synthesized rods, and in the FE-SEM we could see, and EDAX also clearly shows that no other material is found. And zinc sulfide, the sulfonization of zinc rods is very well done, and those images could be seen. And the selective decoration or the limited loading of nickel is very much seen in the third figure on zinc oxide ZNS composite. And the TEM also we have taken, and the rods are clearly seen, and their interplanar distance between those planes are also seen, that the heterostructure is being formed, not that randomly they are separated and they're existing, but rather heterostructure is being formed with a heterojunction. We also have done the PL. The importance of PL and the UV is this: the UV harvests the light, and we can say that nickel is harvesting more light, therefore it may be useful for photocatalytic work. But whereas in PL, the higher the intensity, higher the recombination. The lower the intensity, the recombination rate is rate of photo-induced electrons and holes is less. And coming back to TCS PCA, also we have time correlated related. We have given how much time does the excited electron stays in its excitation state. We have seen that the composite material stays for 3.3 nanoseconds. Therefore, it stays for a longer time, where it can participate in the photocatalytic activity. And we also taken the LSV, current density, where we have found that the composite material has the highest current density, which means to say that it's not just in the application part of that our material is able to do well in photocatalysis, but rather we also want to tell that it is able to transport the electrons when the light is induced. And coming to the transient photocurrent, we could very well see that when the light is on and off, composite material with nickel sulfide is showing highest amount of photocurrent. And we also taken the Motsotsky, where we will know where exactly the conduction band and valency band can be known from here, so that we know our material where exactly its oxidation potential is there, where its reduction potential is there. And also we have taken elemental analysis of how well our material is being decorated. Coming back to the hydrogen production results, we have done on the xenon arc lamp for 4 hours continuously for the various catalysts. and we found the particular catalyst ZNO, ZNS, NIS2, giving 8,471 micromoles gram per hour. And we have done the recyclability also, and we have changed the sacrificial electron donor also. We have used ethanol, methanol, and triethanol amine, but we have found that Na2S and Na2SO3 doing a very providing showing us as a CD, a good SCD. And this is the comparative study that being presented, where ZNO with various compositions, M1, MOS2 or MN dope or the others, our catalyst seems to be doing good, giving a high hydrogen production of around 8.47 micromoles per gram per hour, and apparent quantum efficiency is around 24%. These are our publications. Yes. Thank you. Any questions?
Prabhuji, it's
yes,
good.
sir.
Can you go back once that publication slide? Yeah.
Yes, sir.
Yeah, here. General of Physical Chemistry, General of Nanostructures. Yeah. I have few questions regarding this, because as we are working on this composite materials, which is covered by BRNS. First thing, you have prepared a composite material, zinc oxide, zinc sulfide, and nickel sulfide.
Yes, sir.
I know the role of each one. The zinc is common between the two, and why you have chosen zinc oxide and zinc sulfide only? There are so many metals, no elements, no.
Sir, my whole work, if you see the publication, my whole work is around zinc oxide. So therefore, I have done zinc oxide with copper sulfide. I have done with zinc oxide with copper oxide. Now again, I wanted to see more number of electrons being transferred from the higher to the lower section, which means reduction. If you see the particular, so if in the excitation state, the TCS-PC, if you could see, the longer time they go up and they don't remain there. They need to come down. The moment when they are coming down, that is the time, since the heterojunction.
Band gap between this HOMO and LOMO?
Yes, sir. I have presented here.
Yeah. Amrin madam, is there any time bound for us? Any restriction? Dr. Amrin? Yeah, Prabhuji, please.
Yes,
so,
sir.
Is there any time restriction for our discussions? Time bound?
Yes, sir. 2 to 3 minutes after the discussion.
Then only one more question, because we have to give the opportunity for the audience also. Instead of asking me, it's better to give the opportunity for audience. Okay, last question, Mr. Prabhuji.
Yes,
why
sir.
you have used yet is the symbol in between composite material? Is
at
there
the rate,
yet.
At the rate, it's only a notation, sir. Nothing to do. But rather, if you see the TEM, that sixth one, they're forming a heterostructure.
Okay.
Heterostructure. I want to say they're not isolated and they're not individuals. They're all well knit together.
Then here you have chosen two techniques. One is FESM and HRTEM.
Yes, sir. Simultaneously, I'm showing the morphology, SEM and TEM, sir.
Yeah. Okay. Thank you. Due to time restrictions, I want to leave it to the audience. Audience, do you have any questions, please? Do you want to pose any question to Tarak? If no questions, then we can proceed to next presenter, madam.
I think we'll proceed, sir. If it is there, they may post it.
Yeah.
We'll move on to the next presentation.
Prabhuji.
Thank you, sir.
I would like to now invite Ms. Shahjahan Sheikh. She is a research scholar in Department of Chemistry, Usmania University College for Women, Hyderabad. Over to you, ma'am. Sir, may I request you to please stop sharing, sir? Prabhu, sir. Ms. Shahjahan?
Yeah.
Okay.
Am I audible, ma'am?
Yes. Could you please share your PPT?
Yeah. Is my PPT visible, ma'am?
No.
No. Okay, ma'am. 1 minute. Is it visible now, madam?
No.
Is it visible, ma'am?
No, no.
No. 1 minute. Ma'am, is it visible now?
Yes.
Thank you.
Please make it presentation mode.
Yeah.
Yes.
I am Shahjahan from University College for Women, Kochi. Today I am delivering my oral presentation on eco-friendly synthesis, characterization, and biological evaluation of isochromine derivatives. So isochromine here, they are the heterocyclic compounds with various pharmaceutical activities. For example, if we see the drug sumatriptan, it is a drug which is used to cure headache, migraine, but it was producing a cardiovascular side effect. So later, a drug by name isochromane 6-carboxamide was discovered, and it has not shown the cardiovascular side effects, and it's a safe drug which is used for the treatment of migraine, and therefore it serves as an interesting building block in synthetic chemistry. Later, if we see the natural products like penic isochromane and utsorane, so in that you will be having an isochromane moiety attached to a pyranol moiety, and that is essential for the toxicity of this compound towards human colon cancer cell. Then later, because of these all approaches, the synthesis of different isochromane has taken place. In this approach, Samuel and others has developed synthesized an isochromane compound via the acid catalyzed dehydration of glycol. Simply by the dehydration, the substituted isochromane was synthesized. Then later, another approach by Pates, which will be here the starting material, the scientist has taken isochromane, and after the reaction was carried out, the product formed is dihydroisoquinolinium salt. This dihydroisoquinolinium salt is used in many ionic liquids, which is one of the application of the green synthesis. Then according to whose approach, there is a formation or synthesis of various amides and various benzylic compounds. Here, the benzylic carbon in this moiety is prone for CH functionalization. Then later, according to the Junli approach and according to Kirch approach, there is a formation synthesis of different isochromoin moieties as well as isoquinoline moiety, where it is formed by simple copper or iron catalyst. Then later, according to the Nergis approach, they have taken the aldehyde and reacted with the different aromatic amines. This earlier in the earlier reactions, they have got the product only isoquinolone, but they have used dichloroethane there as a catalyst. But here in this approach, without using the dichloromethane, just if we reflect the reaction in presence of acetic acid, there is a formation of two products, two heterocyclic moiety. One moiety is with isochromane, and the other moiety is with isoquinolone. So here, for the first time, the novelty in this reaction is for the first time in the year 2017, this moiety has been discovered. And here we are getting the two different products, and this the derivatives of these products has been discovered. Then here the proton, these two compounds were separated by column chromatography. The major product formed here is isochroman derivative. Then these compounds was characterized by IR, NMR and mass spectral data, and later this compound was evaluated for antimicrobial activity and antibacterial activity. The antibacterial activity has shown a good zone of inhibition towards the standard drugs like this norfloxacin and ofloxacin with a standard concentration of 1 mg per ml. Then later here, whatever the other product formed in minor amount is also a natural product, is present in most of the natural product, and it is an interesting building block in medicinal and organic chemistry. Then later, different substituents, derivatives of this isochroman as well as isoquinoline derivative was observed, was formed and characterized. Then after this, to a conclusion, here, whatever the products, these two products, whatever has formed, this product has formed by simply reflexing, and it is formed in only one point synthesis, and the result yield was good, and there is a no tedious process here. So therefore, with all these approaches, we came to a conclusion to apply it for further green chemistry. And this is the proton NMR spectra of this isochroman derivative, and this is the proton NMR that spectrum of isoquinolone derivative. And then in continuation, this conventional synthesis, it is our plan to synthesize the compound by greener methods. And I would like to thank Dr. Amruta Madam, Principal of St.Ann's College and co-convener of this conference, and organizing committee members. I thank the co-author and Dr. Narjis Madam and Dr. Alia Madam for this valuable suggestion. And today's Madam, am I audible?
Yes.
So today's special day is National Vaccination Day, and on this day, I wish all of you to pray for a safe coronavirus vaccine. Thank you, one and all.
So
I would like to
some
hear questions from participants. Please, can you pose a question?
So we got a question, but it is for Mr. Prabhu. So maybe we will address it after this.
Yeah. Shajanji,
yeah.
What are the instruments you have in your Koti Women's College to confirm these synthesized molecules?
Sir, we have IR, we have UV, and we will be giving the compounds to HCU, or we will be giving it to campus, sir, and we will be giving in CFRD in campus, sir.
Yeah, the molecules what you have shown are synthesized in your laboratory, the derivatives?
No, sir, we are synthesizing it by microwave in our, green synthesis in our college, sir.
Okay, do you have domestic?
Yeah, we have microwave, sir. We have green method technology.
Is it a domestic microwave oven or that ChemDiscover mode?
Normal conventional method only. Normal microwave only, sir.
Are you using domestic microwave oven?
Domestic only, sir. We are using domestic only, sir.
Then how can you control this temperature and other parameters while doing the reaction? If it is domestic microwave?
By power control, sir. By applying the power mode, sir. By changing the power mode.
Okay.
Power mode is there in our microwave, sir.
Okay. Okay.
Power control is there.
Yeah. Thank you, Z.
Thank you, Ms. Shahjahan. Thank you, sir. Next, I would like to invite Ms. Astha. She is a research scholar from Department of Chemical Engineering, MVJ College, Bangalore. Ms. Astha, over to you.
Yes, ma'am.
So
thank you, ma'am.
Participant from MVJ College. Am I right, ma'am?
Yes, sir. MVJ College, Bangalore.
Yes, ma'am. Thank you, ma'am. Am I audible?
Yes.
Yes, ma'am. Let me share my screen. So is it audible? Is it visible, ma'am?
Yes. Please make it presentation mode. Yes, please. Go ahead.
Okay. So good afternoon, everyone present here, respected jury, fellow participants, and the audience. My name is Astha. I'm a final year BE Chemical Engineering student in MVJ College of Engineering, Bangalore. Our group Our group has worked on synthesis of bioplastic using keratin extracted from poultry-grade chicken feathers, and which will this topic will be presenting today in the conference under the guidance of Mr. Samrajes, Associate Professor, Chemical Engineering Department, NVJ College of Engineering. So moving on, this is the content outline that I'll be presenting to you people. So ma'am, beginning with the plastic, we are very familiar with the word. So plastics are wide range of synthetic and semi-synthetic materials that consist of polymers and are easily available and are cost effective, and also have possessed many properties such as longer shelf life, durable, flexible, and cost effective, and also are very lightweight for application. But since the plastics have been introduced in the market, and continuous usage of plastics have proposed various ecological issues such as accumulation of plastics in water bodies and creating pollution, not only for and also create having harmful effects not only on human beings, also for animals and marine life, and the overall ecology. So to substitute the conventional plastics that are available in the market, we our group went for using keratin. Keratin is a protein available which can be extracted from chicken feather to produce a bioplastic, that is biodegradable plastic, to make a cost effective and easily degradable plastic, so which can be used. So keratin are present in the form of hair, feather, horns, and hooves, and they are broadly classified as hard and soft keratin, depending on the percentage of sulfur present. So, sir, for the source of keratin, we chose chicken feather because chicken feather are a major byproduct, or you can say a waste product, which are generated in poultry farms, and they have almost 90% of keratin content, which can be easily extracted using simple chemical reactions in the laboratory. So for the main objective or the problem statement that we considered for our project is to synthesize the biodegradable plastic using keratin, which is obtained from chicken feather, and also we wanted to do a comparison between the bioplastic obtained from keratin and also the conventional plastics polymers that are available in the market. So we compared the thermal, physical, mechanical, and chemical properties of the biodegradable plastic that we synthesized. So moving on towards the material method, these are some of the reagents that were used. So the first step of production of this biodegradable plastic was pre-treating the raw material. So the chicken feather was locally obtained from a nearby poultry farm, and the feather waste was washed several times using water and neutral soap solution and sodium chloride as the bleaching agent to remove the residual blood and other pollutants that are present in the raw material. Then this clean feathers were laid out on the iron sheets and dried under sun for 3 days. Then this dried feather was milled and chopped into pieces and finally ground to get a powder form that we call it as chicken feather meal. Then we moved on towards extraction of keratin protein for synthesis of the bioplastic. So, ma'am, here we used
there are two methods that were available. One was wet process and another one was dry process. We went with the wet process for preparation of bioplastic. So out of the chicken feather meal that we obtained, we measured some 10 gram of chicken feather powder, and we mixed it with 100 ml of 0.5 ml 0.5 molarity solution of sodium sulfite and 100 ml of 2 molarity sodium hydroxide solution. This mixture was kept in a magnetic stirrer for 2 hours at, at a controlled temperature of 30 degrees Celsius and stirred continuously. Then the mixture that was obtained finally after stirring was filtered to remove some undissolved feather particles that were still present, and also it was sent for centrifuge at 10,000 rpm to separate the supernatant liquid and the solid that was precipitated. So this solution was again filtered using Wattman filter paper, and into that 2 normality of HCl hydrochloric acid was added to reduce the pH to about 4 to 4.8. Then we wanted to increase the mechanical and thermal property of the synthesized bioplastic. Therefore, we added little bit measured quantity of glycerol solution to the supernatant. So the final solution that was obtained consists of your extracted keratin precipitate in the solution. So this solution was again transferred to a petri dish and heated in a hot air oven at 50 degrees Celsius for an hour to get the keratin powder, which is a further process to get your bioplastic. So, ma'am, the final yield that we found out from the raw materials and everything after the process was around 75% of the keratin out of your total 90% that is usually available from raw material. I have included some pictures. So this is the final synthesized biocomposite, that bioplastic that we got after filtration process. So, ma'am, then we further went on to the mechanical properties and the methods that we used to determine the mechanical properties of the synthesized bioplastic. So, ma'am, first one was tensile strength. Tensile strength was determined using the spring test machine. So, ma'am, according to the glycerol percentage that was included in the bioplastic, so the tensile strength were all measured. And similarly, ma'am, we used Young's modulus method to determine the tensile strength. So these are the data that was obtained finally after the test. Then moving on, ma'am, we also measured the film thickness. So, ma'am, from the glycerol percentage, that is 2%, 5%, and 10% glycerol amount that we used for preparing the bioplastic. So the thickness was determined using the screw gauge, and the same film thickness we used for determining the properties of polyethylene and polyesters that are conventional plastics that are available. So, these are some of the data that was obtained. Then, ma'am, further again, mechanical testing that was done using the same Young's modulus and mechanical testing machine equipment according to the ASTM standards. So, ma'am, again, we compared it with the polyester and polyethylene. So these are again some of the data according to, so we also calculated the cross-sectional area, the tensile strength and elongation, and we compared all the plastics, ma'am. Then, ma'am, water absorption is an important property that needs to be verified when we are synthesizing any plastics. So, ma'am, these are the data. So we took 2 to 5 grams of the synthesized plastics and we immersed in water for 24 hours and 48 hours. So, ma'am, the percentage of percentage water that was absorbed by the plastic is shown in the table. Then finally, ma'am, since we are preparing biodegradable plastic, so obviously degradability test is also important. So, ma'am, the sample that was obtained, it was kept in soil surface for 5 days to monitor the degradation level of the bioplastic. So, ma'am, this is the data that we obtained. It It's giving the quantity that is lost during the degradation in the period of 5 days. So finally, I would like to conclude my presentation by saying that we conducted this experiment to synthesize the biodegradable plastic using chicken feathers, and from the literature survey that we did, we were successfully able to get like good percentage of keratin from the raw material, and we were able to synthesize the biodegradable plastic. And also, after doing the mechanical and thermal property analysis and other analysis, we found that our plastic that was synthesized has shown better properties compared to your conventional plastics that are available. And also, in terms of cost effectiveness, the raw materials that were used were basically the waste materials that we are converting it to a useful material, which can be, if possible, it can be commercialized in future also. So these are some of the references. I'd like to thank your institution for giving me the opportunity to present my paper. So with this, I'd like to conclude my presentation. Thank you.
Yeah, Miss Asta, the title is so interesting. First
Thank you, sir.
of all, when did you register for your PhD?
Sir, I'm a student currently, final year.
Final year. That means yeah, MTech are you?
No, sir. BTech.
BTech. Okay. Then how did you get this idea?
Sir, actually, we were searching in this field only. Our aim was to find some alternate. We heard about alternates of plastics and recycling of plastics, but better than that, why not go for any biologically available
Is
plastic
it part of your
manufacturing?
internship? The project what you are doing?
Sorry, sir.
Is it part of your internship?
Not exactly, sir. We took it as a mini project, you can say.
Yeah. Okay. Thank you. Thank you,
sir.
Astaj.
Thank you, sir.
Any other questions from the audience? So I think we'll move on, sir. Last presentation of today's session is by Ms. Lara Kehkasha. She's from the Department of Biochemistry, St. Anne's College for Women, Hyderabad. Ms. Kehkasha, over to you.
Good afternoon, and all present. Am I audible?
Yes, but please be a little louder.
Okay, just a minute. I'll try to authentic. Am I audible now?
Yes.
Okay, thank you. Good afternoon to one and all present. Today I, Lara Kehkasha, would like to present a paper I co-authored with Ms. Sushma, HOD of Biotech Department at St. Anne's. The title of our paper is A Review on Biodegradation of Microplastics. Plastics. We all know about them. We may not love them, but we use them every single day. By 2050, researchers estimate that there will be more plastic than fish in the ocean. At least 350 to 400 million tons of plastic are produced yearly. Plastics are further broken down into the smallest components, microplastics, which are known to cause adverse effects and is a huge concern for healthcare. Pollution caused due to microplastics has been a rising environmental crisis because of their persistence and varied effects on ecobiology. The animation here shows how a plankton has consumed microplastics present in the ocean in a considerable amount. This is just to highlight the gravity of the situation. The green highlighted part is fluorescent dyed microplastics, which are found in the gut of plankton. So how did we end up with so much plastic waste? Well, plastic is cheap, durable, adaptable, and it's everywhere. But the good news is that there is something else as well, which is extremely cheap, durable, adaptable, and everywhere. And research shows that it may even be able to help us with our plastic problem. I'm talking about microorganisms, specifically bacteria. Microorganisms are known to adapt and survive in the midst of very adverse and harsh conditions. Microorganisms possess an inherent ability to adapt to almost every environment and can also degrade microplastics. The potential of microorganisms being used to degrade microplastics is a very great and promising strategy. Our paper focuses on biodegradation as a means to reduce microplastics. Biotic degradation usually occurs due to the action of microbial enzymes. It converts organic compounds into simpler organic compounds such as carbon dioxide, methane, water, etc. Bacteria and fungi are the most important types of microorganisms in the biodegradation of natural and synthetic polymers. This is because biodegradation depends on various environmental factors as the biodiversity and occurrence of microorganisms vary accordingly. Microplastic degradation is an integrated process of physiochemical and microbial degradation factors. Since microorganisms can adapt to every environment and have potential to degrade microplastics, they can be used in bioremediation. In the environment, microorganisms are in contact with microplastics, organic matter, and inorganic particles. This increases their adherence to the surface, which acts as a substrate and leads to the formation of favorable conditions for creation of biofilms. Biofilms are collective of one or more types of microorganisms that can grow on many different surfaces. Since the creation of biofilms is characteristic of biodegradation, the main microorganisms focused upon in this paper were bacteria, fungi, and protists. Thus, this enzymatic degradation leads to the loss in characteristic properties and structural damage. Bacterial strains catalyze metabolic reactions that are attributed to microplastic absorption, desorption, and breakdown. Bacteria were specifically and extensively researched since they were able to grow in minimal nutrient media. They use polymer materials as the sole carbon source, reducing the dry weight, average molecular weight, and molecular distribution of polymers. They bring about various morphological and chemical changes in the structure. This leads us to think that maybe these bacteria can be used for the reduction of microplastic pollution in the environment. Microplastic degradation was studied in 2 bacterial consortia, that is 2 or more bacteria living symbiotically. This was done so that it could be seen how they might react in a naturally occurring biofilm. The use of bacterial consortia eliminates the effects of toxic metabolites produced by some strains present in the consortia. This was the data obtained by a different study journal regarding the bacterial strains capable of degrading microplastics under laboratory conditions and in the environment. It is very important to note is that the least amount of degradation taken was 8 days. Out of all of these data, it is the least amount of time taken to degrade microplastic was 8 days, and it was done by Comamonas acidovorans. It was seen that the process of bacteria eating plastic is actually a natural one, but it's a very slow process as visible. A lot of research needs to be done to speed up this process to a useful pace. Biodegradation by actinobacteria. Actinobacteria, which are commonly known as actinomycetes, are gram-positive bacteria. These are present in varying ecosystems such as marine systems and various other ecosystems. Besides the production of antibiotics, anti-cancer drugs, fungicides, some can digest resistant carbohydrates. Others are used in bioremediation due to their ability to degrade toxic materials or play a role in the degradation of complex polymers. This was the data obtained with actinomycetes. Actinomycetes have relatively good degradation capacities of various microplastic types, which is visible in the table. Bacterial consortia with actinomycetes and different bacillus species presented a high capacity of biodegradation, which is why it was presented as a different study and not in continuation of bacteria. The highest polymer reduction percentage was found in mesophilic mixed bacterial culture. The actinobacteria in the table show efficient degradation capacity of PLA as well. These can degrade PLA, polylactic acid, under lab conditions. Biodegradation by fungi. Fungi are the best suited candidates for research of their capability in plastic biodegradability because of their vast metabolic pathways and potential, which includes the extracellular multi-enzyme complexes. As seen with bacterial species, microplastic biofilms are a great source of carbon and a cultivation ground for fungal species that can degrade plastic fragments. More importantly, biofilms act as a hotspot for microbial activity, such as fungal and bacterial growth. This was the data obtained for fungal strains capable of degrading myoplastics under laboratory conditions and in the environment. The composition of fungal communities on biofilms differs significantly from surrounding environments. This is a very important point because it can be both good and bad. This is because it might be an opportunity for finding novel plastic degrading metabolic pathway or species, or it might also prove to be a source of pathogenic species and pose a health concern. Fungi overall take significantly less time to degrade microplastics than bacteria or actinomycetes. In researches conducted for biodegradation of plastics and microplastics, fungi are often excluded. Thus, there is a lot of potential to be found here. Thus, to conclude, biodegradation of microplastics is a potential solution as several microorganisms are capable of degrading microplastics. Bacteria, actinomycetes, and fungi are present in various ecosystems, including marine, and have several distinct metabolic pathways, which provide great potential for biodegradation. Plastics and microplastics were seen to be efficient soil carbon sources in a culture media. So what next? It is now necessary that after all these researches, new sustainable and cost-effective synthetic materials and methods are devised. Marine ecosystems need to be extensively researched for targeted isolation of new microorganisms capable of microplastic degradation. There is also now an urgent need for papers and for scientists to focus on the solutions to combat pollutions caused by plastic and to find sustainable degradation methods. Thank you very much for your attention.
Yeah, Miss Laraib, what is your present status?
So I'm a second year BSc student.
Okay, BSc second year students. Who have provided this information for you? How many papers?
So around 25 papers were chosen, but specifically 3 papers were focused because they were the ones that provided data with fungi, bacteria, and actinomycetes data for.
Can I give one small suggestion?
Yes, sir.
It's a suggestion only, positive suggestion. In future also, don't read any slides, okay? We can also see, we can also read the slide. Just we are looking for your explanation. Okay, it's a small suggestion.
Yes, for sure.
Dr. Amrin?
Yes, sir.
Still any presentations?
Any other question? No, sir. This was the last one.
Yeah.
I'll hand over it to Ms. Ramya to please conclude the session.
Yeah, and before that, I would like to thank our principal and Dr. Ammani Das. She has given an invitation to me. So I'm very much glad, madam, to be part of this international conference. And I thank Dr. Amreen as well as and the organizing committee who are part of this international conference. Thank you. Thank you so much.
Thank you, sir.
We are also privileged, sir, that you have come here. Thank you so much for that.
Thank you so much, sir. Ms. Ramya, please.
Now I call Nazia for proposing vote of thanks.
Thank you, Ramya.
Over to you.
Thank you, Ramya. So, with the successful completion of our technical session 2, Here we have come to the end of the session 2, day 1 of our conference on emerging trends in medicinal and green chemistry, ETMGC 2021. I once again thank all the dignitaries for gracing the occasion. On behalf of Department of Chemistry, St.Ann's College for Women, I thank our esteemed speaker of the session 2, Dr. Kalpana Madgula ma'am, Research and Development Chemist, SAS Nanotechnologies, Wilmington, USA, a keynote speaker for her valuable insights on green coatings and her contribution towards greener approach, which is much needed for the sustainable future. Followed by the technical sessions chaired by Dr. K.N. Katari sir. We thank you, sir, for sparing your valuable time with us and accepting our invitation to be a chairperson. A big round of applause to all the presenters for the great work presented in the field of medicinal and green chemistry. I would like to thank Dr. Sister P. Amruta, Principal, St.Anne's College for Women, for her constant support, and Aparna ma'am, Technical Director, ICT. Before I conclude, may I request all the participants to fill the feedback link of session 2 posted by Ms. Deepti, Assistant Professor, Department of Chemistry, in the Zoom chat box, as well as in WhatsApp groups. As I have mentioned earlier, there are 2 feedback links, one feedback link for presenters and another feedback link for the participants. The feedback link will be active for another hour, that is 60 minutes time. All the attendees will get in order to fill the feedback link. And after that, the feedback link will be deactivated. Thank you all once again, and special thanks to our sponsors, Union Bank, SP Acure Labs, Touching Lives Globally, and Jolly Scientific Traders. So let's connect with the same zeal tomorrow on Zoom using the link at 9:45 a.m. sharp. Thank you all.
8:45.
Nazia, next laptop? Huh. No, I'm on the phone. Yeah.
Should we end the meeting, Nazia?
Amreen, I'm going to end the meeting now.
Okay.